off, or dried and dies off, and then next year new leaf from a new rhizome comes up, sometimes along with an inflorescence, or sometimes inflorescence later on. Okay, then there are another type of morphological diversity is the terrate leaf, like needle-like leaf. Okay, this is a genus called Lucia. Lucia, we have about 4 to 5 species, and this is one among the species, Lucia zeylanica. And you can see the leaf, it is roundish in shape or cylindrical in shape. Okay, so then we have myco-heterotrophic trophic, orchid, which do not have chlorophyll, and they solely depending upon soil mycorrhizae for their what do you say, food necessities. Okay, so they are also called saprophytic fungi. Now people prefer to call it as myco-heterotrophic orchids. And then there are orchids with the bulbous base to overcome hard conditions like tough conditions, so they become xerophytic. And also in the leaves, you may see sunken stomata also with the hard cuticularized epidermis. Okay, some very unique features of orchids are presence of Polynia, then presence of Polynia like this, like pollens are aggregated in the form of mass, and then the presence of a fused stamens and pistil called column or gynostigium, and then the non-endospermic seeds. The seeds are very, very minute, and that's why they comes under microspermae. Okay, then you can see very unique pattern of morphology, structure, arrangements of floral organs, like petals, sepals, especially in petals, the lip do have a very wide variation. Okay, then some other general aspects. One of the most diverse family or species family with approximately about 25 to 30,000 species. The number varies in different references. So maximum up to, there is no compiled checklist also so far, and about thousands or 10,000s or more than that of varieties, cultivated varieties, and still people are coming up with new varieties. Okay, then it includes terrestrial as well as epiphytic species for even for cultivar varieties also. Okay, some species are mycotrophic, this I've already told. Okay, we have, I mean, there was no precise classification existed earlier. And I mean, since the diversity or the number of species were too high, the taxonomy is very difficult, because rather than differences, here you get most maximum similarity. The dissimilarity between two different species is very minute, so it makes the taxonomy or classification or categorization of species is very difficult. Okay, so but Robert Louis Dressler, we have to thank this particular person who dedicated his life for classification of orchids, and he could completed his work by 1981. And between 1981 to 1993, there were few revisions and additions happened, and by 1993, the orchid classification is almost perfect. And but still, some additions or some modifications are going on. Okay, so according to the present classification, the family Orchidaceae is divided into 5 different subfamilies: Apostatsioideae, Cypripedioideae, Orchidoideae, Epidendroideae, Vanilloideae. 5 subfamilies. I will deal each and every one separately. Okay, subfamily Apostatsioideae, the most primitive form of orchids. So I have never seen these orchids, and I don't know, this particular one is not present in India. I think Apostasia, there is one species present in India, maybe in Assam or Arunachal Pradesh. And it is characterized by the presence of 2 to 3 fertile anthers. That means in orchids, there are 1, 2, or 3 orchids. And likewise, I mean, they have classified as monandre, diandre, and triandre. Okay, so different groups of orchids do contain a specific number of anthers. Okay, so here in this particular subfamily consists of 2 to 3 fertile anthers. There may be sterile anthers or staminodes also, but they will be fused to the gynoecium. And it consists only of 2 genera, and this Apostasia. Then there is one more at another species, genus Nuvidia. Nuvidia, this is the Nuvidia species. I don't know from where this particular picture, there was no indication in the internet along with the picture. And there are about 15 species altogether. There are about 15 species under this subfamily. So it's a very small family considering other super subfamilies. Okay, then another very interesting subfamily is Cypripedioideae, and with a common name called the lady slipper orchids. And almost all the species or all the genera comes under this subfamily do have a very inflated or pouch-shaped lower lip, and which is analogous to the lady slipper. That is why the name came. About 150 species of, or now, I mean, a recent record states that 165 species, and almost all species of Cypripedioideae are terrestrial, and they consist of 2 fertile anthers. Okay, and with a shield-shaped stamen, you can see here is shield-shaped stamen with a wing-like external appendage, and then a pouch-shaped lip. These are the characteristic feature of Cypripedioideae. Okay, then some of the genera Cypripedium. This particular picture that you are seeing is a Cypripedium. And then there is Paphiopedilum, and this species is endemic to Kerala and Tamil Nadu. Paphiopedilum drury, which is a restricted species, a scheduled species. Okay, then Phragmipedium, which is, I think, temperate. Phragmipedium is also considered as you look at the lateral petals, the length of the lateral petals, very unique and very rare orchid, I guess. And then Mexipedium, you can see that I mean, this is also very rare with very small flowers, but look at this small flower with a micro lens. Through a micro lens, how beautiful it is. And it is very, I mean, the arrangement of floral organs are very, very unique from all other groups in the lady slipper subfamily. And then Selenipedium. Selenipedium also do have a very unique lip, which is folded inward, and then very narrow thread-like lateral petals. Okay, then the subfamily Orchidioideae. It is a subfamily with lots of species which have a single fertile erect anther. So it is a group wherein the androecium is monoanthous. Okay, then the species of orchids comes under Orchidioideae are very, very interesting. This is a bee orchid, which is Ophrys, wherein people have studied the deception, sexual deception in orchids, wherein this particular lip mimics exactly like that of a female bee, and also it produces the pheromone exactly like that of the female bee produces. And in illusion, the male bee comes to this particular lip, thinking that it is the female, and it tried to mate with the lip, but it goes in vain. And in due course, it may collect the Polynia and then transfer it to the next what do you say, the lip or labellum, wherein the bee find a pseudo female. Okay, so then there is Habaneria. This is Habaneria diphylla, which is distributed in the Western Ghats. Then Satyrium nepalens, it is distributed in India, Sri Lanka, and Nepal. So then Spyranthus. Spyranthus is a very unique marshy species, in which the inflorescence is spirally twisted. Okay, then there are so many other genera comes under Orchidioideae. So I have selected some of the genera to have a understanding about the structure. Okay, then the fourth subfamily is Epidendroideae. So this is one of the largest subfamily, which consists of more than 80% of the total orchid species comes under this particular subfamily Epidendroideae. And it also consists of single anthers. That is sub-erect. Sub-erect in the sense, in all other orchids, it may be very straight and standing upright. But in this case, it may be growing, or it may be bending towards the outer edges, little bit of a spreading effect. Then it is the most widespread subfamily represented with more than about 80% of orchid species. And you might have seen this, these are not cultivated varieties, and these are wild species, so there might be some differences. This is a wild Phalaenopsis, Phalaenopsis amboyensis, and then Phalaenopsis chibae, then Oncidium, the dancing lady orchid. So there is, I mean, you may find a little bit of differences from that of the cultivated variety. Okay, so this is Oncidium, and this is also an Oncidium, Harrisoniana. Okay, then Dendrobium, so which is a very large genera consisting of hundreds of species or thousands of species all around the world. It is mostly distributed in the tropical Asian region. Okay, then Tropidium, which is a very rare orchid in the evergreen forest. I have found this in some places in Karnataka and some places in Idiki district. I've seen it in a cardamom plantation in Idiki, but no one could believe that this is an orchid unless you see the inflorescence. But this inflorescence doesn't have a flower, and so far I haven't seen any flower in this particular plant. And right since the first day itself, I was doubtful about, by looking at the leaf, I was doubtful about whether, I mean, it is an orchid. Okay, then Vanda, another very common familiar name for us. This is Vanda thwaitesi, and then Vanda testacea. This Vanda thwaitesi and Vanda testacea is widely distributed. Vanda thwaitesi is a Western Ghats species. Then the last subfamily is Vanillioideae, which is very commercially very important, and it's okay. It's also a monandrous orchid. So most of you are very familiar with Vanda, but I'm not. Sorry, vanilla. Okay, but I don't know whether you are familiar with this particular vanilla, which is distributed in the Western Ghats and in some other places also. So it doesn't have proper leaves. Instead, there are scaly leaves. And this is called Vanilla ephylla, or with the synonym Vanilla whitiana, and with a very nice flower, but the color varies from place to place. And that is not only applicable for Vanilla ephylla. The color of the orchid species may vary from one place to the other, depending upon the local and regional climatic condition. And other than vanilla, there are Cleistus, then Certosia, and Lecanorchis, then pseudo vanilla. So if there are no flowers, then you might consider this as vanilla, but there are differences in the floral organs, and then it is considered as a pseudo vanilla. Okay, then let us see what are all the characters that you have to look at when you consider about the taxonomy of orchids. So you have to consider whether an orchid is, first of all, whether an orchid is perennial or short-lived. There are annuals, there are biennials, there are perennials. So most of the epiphytes are perennials, and but whereas most of the terrestrial forms are annuals. But you might be able to see leaves coming up each and every year at the same place, but that is because of the lateral branching. Okay, then whether it is terrestrial, epiphytic, lithophytic, or saprophytic, or mycoheterotrophic. Okay, so this is a perennial orchid, and this one is a lithophytic orchid that is growing on a rocky substratum. And then this one is growing as an epiphyte, but sometimes you might be able to see orchids are still thriving on dead branches. Until the branch fallen down, the orchid can survive. Even after fallen down, they can survive for some time until they get any kind of serious infection or water lodging problem from water lodging. And there are autotrophic or mycoheterotrophic herbaceous plants. These are also annuals. So there are about 45 species of mycoheterotrophic species in India. Okay, then most of the terrestrial forms are having either rhizomes or tubers or rootstocks. So here is an example. This is a Nervilia, and this is a Nervilia prainiana with flower. This is Habaneria bractescens. Okay, yeah, this is another Habaneria. Then there are sometimes some of the orchid, then you have to consider these two terminologies, like a sympodial. This one is a sympodial orchid because they are seen or growing as clusters. This is one branch, and another branch comes from adjacent to the maternal branch. And first year, this will be flowering. The second year, this might be flowering. And then the third year, the other one would be flowering. And only one or two leaves from one bulbous base or one bulbous petiole. And the stem will be very short. And this is a monopodial orchid because it do have only one branch from one place or from the substratum. Okay, so yeah, this is a little more clear. A sympodial orchid will have a collection of branches, but this will have mostly one branch at a time. I mean, from a place, substratum. Then orchid stem is very, very minute. I mean, some of the orchids do have like what is Corymborchis, do have a very clear stem and internodes. But these are pseudostems, bulbous pseudostems, porpax. Okay, then this also, this is Concidium, and this do have a very bulbous pseudostem. Here also, again, porpax. Yeah, then, but Dendrobium do have a very stout, erect branch. Some of the Dendrobium do possess leaves when in flower. Some of the Dendrobiums might not be having leaves when in flower. That depends upon which time they flower. Most of the summer flowering orchids do not have leaves while in flower, but most of the rainy season flowering plants or Dendrobiums do have leaves as well as flowers. Then there are lots of other forms of orchids. I mean, orchid stems. You can find differences in orchids. This do have a very tiny pseudo bulbous condition, and yeah, there is a very stout, tiny bulbous. And here, this might be the petiole, swollen bulbous petiole, and the stem is this one. Yeah, and likewise, there's a huge diversity in form, appearance, and morphology of habit and leaf structure, things like that. Okay, let us go to the leaf. Okay, look at the leaves. Usually, an orchid might possess 1 to several leaves. Most of the annual flowering plants do have less number of leaves, and most of the perennial species do have maximum number of species, and the number varies from species to species. And there are species which are saprophytic that do not have leaves at all, green leaves at all. There may be some scaly appendages, but there is no chlorophyll at all. Okay, so yeah, this also doesn't have a proper leaf, but scaly leaves. Okay, then this is another one, Teniophyllum. This one doesn't have leaf at all. And the photosynthesis is performed by means of the chlorophyll present on the root. So these are root orchids or leafless orchids. Teniophyllum Cheloschista. There are few species which are root orchids in which you may not be able to find leaves at all. And if you happen to see one branch or this individual, you might be thinking that some poor orchid which is dying. Okay, and this is very hard to find. Most of these orchids, leafless orchids, are found in the top of the canopy, mostly in the canopy in this tiny branchlets. So because they don't want maximum leafy surface for capturing lots of energy, and you can see that their fruits are very small, and the flowers are also very tiny. You can see the remnants of flowers here. It's very tiny. Okay, then there are bulbous based stems. This is Polydota pallida, and then this is Bulbophyllum sterile, which forms a pseudo bulb. The swollen petiole forms a pseudo bulb, which actually help them to overcome adverse conditions or drought conditions. This also do have a bulbous internode. I mean, sorry, this is also a petiole. Okay, the internode is here. Okay, and this is I forgot. I am not getting the name. Okay, yeah, this is Triastoxi. Triastoxi also had. I mean, this is very hard. The stem is very hard. This can be seen little. This is Siolagaine, and this is Triastoxi. This is seen growing little high altitude and not in the lower altitude. Okay. Then there are leaves like this canaliculate leaves, which is folded little bit, and yeah, with aerial roots, epiphyte. Yeah, sorry, this should be root, not leaf. Okay, so orchids do possess aerial roots as well as the clinging roots, which is used for attachment, and then there are roots which is hanging down freely that may or may not contain chloroplast or chlorophylls. Okay, then it another unique feature for an epiphytic orchid is the presence of velamen, one or more layers of velamen in the cortical region, just instead of epidermis. Okay, yeah, yeah. This is the velamen area, the velamen structure of an orchid, and this is the exodermis. It's a little harder, a little yeah. And then this is the cortical region, parenchymatous cortical region with the vascular bundles. Okay, then sorry. Then coming to inflorescence, they may borne basal, lateral, or terminal. This is a plant wherein the lady slipper orchid, wherein the inflorescence is born from the base of the orchid. And here, this is a terminal flowering orchid, which do possess only one solitary flower. So again, this is a calanthe species, which is developing again from base, and this is from the terminal portion, and they may be either erect or pendulous. Okay, then they may be, the inflorescence may be solitary with only one, with a spicate or subumbellate or paniculate. This is a spicate inflorescence. You might be thinking that this is the pedicel. Actually, it is not the pedicel. This is the ovary, inferior ovary, and there is hardly any pedicel in it. Okay, then yeah, they may be arranged very tightly, or they may be very what do you say, loosely. Yeah, these are, I mean, this is a disticus inflorescence, and this is a second inflorescence. Yeah, again another inflorescence. Sorry, this is the second inflorescence, which is laterally one-sided one. Okay, when you come to the flowers, you might come across flowers as large as that of 5 centimeters to 10 centimeters, but there are flowers with 1.1 millimeter to 2 millimeters. This particular Oberonia Chandrag or Oberonia Rangannayana, which is seen distributed in Goa, Karnataka, Tamil Nadu, and Kerala, the flowers are very, very tiny, and it is very hard to take a picture, a macro picture like this. Okay, and this one is again harder, 2 millimeters, Saculabium gemmatum. It is very difficult to even find the plant. But there are very large flowers also. Pectilis gigantea or Pectilis susannae. The across length would be around 10 centimeters, even little slightly larger also. And this is about 2 centimeter to 2.5 centimeter. This is Aridus crispa. Again, an epiphytic orchid. The other one, Pectilis, was a terrestrial orchid. Okay, they're very showy. Most of the larger ones are very showy, and mostly they are zygomorphic or slightly actinomorphic. Most of the orchid flowers are zygomorphic in general, and they are bisexual, sessile, or sometimes pedicellate. Then most often they resupinate in the sense, in the bud stage they will be in the axis, inflorescence axis, like this, facing upward, and then they twist around and facing downward 180 degree, and then open the flower, keeping the labellum downward. In bud stage, in young bud stage, the labellum was on upper side, and then when the flower opens, the labellum comes to the lower side, as usual, whatever you see here. But there are orchids which do not go for resupination, or sometimes some of the orchids, they resupinate 360 degree. I don't know why they are doing so, but this 100 degree resupination or twisting helps them to properly place for the incoming insects or visitors for pollination. So here it explains, this pictures explains the resupination. It was upside, it was pointing upward, and then twisted downward and started opening, and then it opened and keeping the thing. Otherwise it would have been like this. Okay. Yeah, here this doesn't go for resupination. This is opening as if it is there. Okay. Oh. So then again, this is a typical orchid flower that I've chosen from my photographs. This is Flickingeria nodosa or Dendrobium nodosum. These are the sepals, and we cannot, since we cannot distinguish the petals and sepals, and we call this as a perianth, but these are, I mean, we can easily, by the placement, we can easily identify these are the outer sepaloid perianth and sepals or sepaloid perianth, and these are the petaloid perianths, of which there are 3 petaloid perianths, of which one transforms or modifies to form a structure called lip or labellum. And you would be amazing by understanding the fact that how many different types of lip or labellum is there among orchid. I will show you some other pictures later on. And there is another structure, column, wherein the androecium and the gynoecium fuse together to form a single structure, or you can call that as a gynostrigium. Okay, so the placement of this is very important for pollination to happen, and you can see some projections or some articulations in the flower, which actually serve as the pollinator guides to the resources wherein the rewards are kept. So they have 3 plus 3 perianth lobes, 3 outer sepals and 3 inner petals. And one of the petals is transformed to form labellum, which is platform for an insect. Okay, the sepals usually are free, or sometimes they may be variously adnate or fused to the what do you say, the spur or the mid lobe or the laterals. Okay, these are some of the pictures from Dr. Pankaj Kumar. He had described some of the orchid species quite recently, who is working in the Hong Kong Botanical Garden. Okay, yeah, and then there is another one. This is the spur or spur-like mentum. This one. This is a spur. Okay, and there are different types of forms in the arrangement of petals and sepals. And you can see the Cypripedium calceolus, and then Paphiopedilum. You can see differences in the arrangement, shape, and placement of floral balls. And there are n number of variations, n number of floral structures. And here you would find it difficult to identify which are petals, which are lip. Anyway, the lip is inserted inside. You cannot see the lip from outside. You can see a little bit. Yeah, here this is the lip, which is very small. Here the sepals are more attractive, more larger than that of this is Bulbophyllum fishery. And there are so many different species. Yeah, here also so many. This is also a jewel-like orchid. Yeah, all of them are Western Ghats orchids. And some of the foreign orchids wherein you can see lots of differences, and several people attribute that the orchid flowers are mimicking some other organisms or some structures, and I don't know to which organism these flowers are mimicking. Some of the mimicry that people have identified and they have worked out, and most of the mimicry are meant for attracting their insects, and most of the orchid do not offer any rewards. Okay, so you can see lots of different forms of lips here. Here also, something which is flying bird or something like that. Yeah, here this is the Indian bee orchid Cotonia peduncularis, wherein exactly a bee, I mean, it is the lip or labellum is mimicking a bee. Unfortunately, so far nobody could identify which bee it is. Yeah, here also another view of Cotonia peduncularis. Then a monkey orchid. Monkey orchid is very attracting. It looks like, I mean, a face of monkey. And then another one, bee orchid, that I have already shown. A group of bees together forming a colony or something like that. Again, some other insects sitting, a sitting posture of some other insect. And there is something else. Yeah, and also swaddle babies. You can see that somebody has kept babies inside and the baby is sleeping inside. And then a flying duck or swan, Calina major, it exactly looked like that of a swan which is about to fly or landed. Then another important structural feature of an orchid is the column. Column or gynostegium might be short to long, and the size may vary depending upon the proportionate size of the flower, which consists of anthers from 1 to 3 numbers with an anther cap, a protecting anther cap. An anther cap is removed in this flower. It is not there in this picture that I have shown. And the number of Polynia may vary from 2 to 8. It may be sessile or attached by stalks. It is technically called caudices or stipes with a sticky visidia at the base. And this is a drawing of a column wherein this is the anther part, and this is the anther cap, and the anther cap has to be removed by the incoming visitor to get this Polynia. And this is the visidia part, the disc-shaped visidia part with a lower antenna, and there is a rostellum. Rostellum is an extension of the stigmatic region, and here is the stigma. This is the stigma, okay, the underneath. And here in this particular flower, here inside is the stigmatic region, which is very sticky, stickier than that of this visidia, so that the Polynia which is attached onto the head or body of the insect will get attached onto the stigma when it comes in contact with the stigma, because it is stickier than that of this visidia. A stigma might be three-lobed with the mid-lobe often modified to form this rostellum. Okay, other lobes are either sunken or they forms the ventral surface, or they may be kept behind the column. Okay, which may not be able to distinguish. You can see a little more detail. And this is a Polynia, another picture of a Polynia, which is very small. Sometimes it is very small, sometimes it may be larger. Again, it might be depending upon the general size of the flowers. Here, the ovary is inferior, where the flower is epigynous with unilocular and parietal placentation. But if it is axile placentation, then it would be a trilocular. So both unilocular condition as well as trilocular condition shall be seen in Orchidaceae. If it is unilocular, then the placentation will be parietal, and parietal placentation with unilocular condition is the common one, and trilocular condition with axile placentation is also present. Yeah, this is an inferior ovary with a short pedicel. So yeah, and this is a fruit, a developing fruit with a mature fruit that splits open. Fruit is usually a capsule, mostly a capsule. Rarely berry-like. I haven't seen any berry-like this thing. Some small ones are there which might be berry-like. Seeds are very numerous. Lakhs of seeds would be there which are microscopic, which doesn't have endosperm, and therefore sometimes they are very rarely winged. And therefore since it doesn't have endosperm, it requires some external source for the development of embryo. Mostly that purpose is served by external fungus. The microtrophic fungus that will provide nutrition for the growing embryo. So many a time the orchid seed has to reach a place where this mycorrhizal fungi is present. Otherwise, it would not germinate. Okay. Then there are so many species discoveries happening in India and in all over the world. And here in India, this is quite recently published paper in 2016, Habaneria sahyadrica by one of my great friend, Dr. Pankaj Kumar. And there are so many other people also involved in the discovery of Habaneria sahyadrica. And you can see how and which are all the structures that are necessary for describing an orchid. And also this is very crucial for running key for identification also. Okay. Then in India, there are about 1,256 species of orchids in India, according to the latest update by the Botanical Survey of India. They have recently published a book on orchids of India. And they claimed that almost 60% of them are epiphytes, with 447 species are terrestrial, and 43 species in India are mycoheterotrophic or saprophytic species. Okay, so I'm trying to give you some clue about how to run the keys for selected genus of Western Ghats species. Okay, and thanks to Dr. Jivan Jalal Singh for providing me this key. Okay, then yeah, let us see. If you get a specimen which comes under Vantoideae, and which looks like Vanta, and you can easily identify that as an Eridus, genus Eridus. But if you want to see the species, then you try to, what do you say, run this particular key. Stem less than 20 centimeter long, then you have to go to 2. Yeah, what 2 says is that stem more than 20 centimeter long. Sorry, 1B, sorry. If it is not 2, then I'll show you the 2 later. So then 1B stem will be more than 20 centimeter long. Okay, if it is 2, that means the stem is less than 20 centimeter, then check whether the flowers are pinkish violet with the mid-lobe of the lip broadly ovate like this. Yeah, the mid-lobe is broadly ovate at the center, or yeah, here. So you can say that it is Eridus maculosa. Okay, then go to the, I mean, if it is not this one in 2, then see whether the flowers are white, then flushed with pink, a little bit tinged with pink, mid-lobe of lip linear. Unlike this one, if where the mid-lobe was broadly ovate, here the mid-lobe will be linear. Look at this one, the flowers are almost, surface color is white with little bit of pinkish tinge, and the lower lip is linear. It's very linear, unlike the previous one. Okay, then if it is not the 2-1, then if you had got a specimen with more than 20 centimeter, then see whether the mid-lobe of the lip very larger. I mean, and this terminology is a little bit confusing. So you have to handle these specimens for a while, then only you can distinguish how you can make it, whether which is larger on a comparative account. And side lobes of the lip much shorter than the mid lobe. Okay, here, yeah, these are the side lobes, which is much shorter than, let us see the previous one. Yeah, here it is even shorter, it seems. Yeah, it is very shorter. Shorter than when compared to the mid lobe. When compared to the mid lobe, the side lobes are very short. Okay, so this is very, and it is not a spreading one. So this size, the side lobes, the size of the side lobe is not a comparative one. It is comparing here with this lip, the breadth of the lip. Okay, and if this condition is agreeable, then it would be Eridus crispa, and if it is not, then run the next one. Mid-lobe of the lip short. Here is the picture of the next one. Mid-lobe of the lip short. Yeah, it is shorter when compared to the other one. This is little enlarged picture. Side lobes of the lip much larger than the mid-lobe. Yeah, side lobes here. It is standing upright, and yeah, it is standing upright. The side lobes of the lip much larger than the mid-lobe. So if you can run this key, this is a very small genera, consists only of 3 species in the Western Ghats. So it is very easy to run the key. Another genus Area, and I will be dealing with the related genera also, so that you can have a comparison. So when you get an Area genus, a species of Area, then run the key, and it is very straightforward for most of the species. If you are getting a species like this with flower about 4 millimeter long, you can imagine that how big or how small this flower is. So the person who had taken this flower might have enlarged quite a bit. Okay, entire flower is 4 millimeter long. Lip with a pair of lateral papillae at the base. Lip, this is the lip with a papillae, maybe this one. Yeah, it is not visible anyway. At the base, then it is Area albiflora. Okay, then if the flower is 1 to 2 centimeters long, yeah, it is quite bigger. Then lip without any papillae, then you can straight away go to 2. And let us see what are there in 2. Pseudobulbous ovoid. So the presence of pseudobulbs, area in most of the area, there are pseudobulbs. Then inflorescence with many flowers or several flowers. And this is the area. Here, the number of flowers per inflorescence is limited, but sometimes in some conditions, you might be able to see large number of flowers. Okay, then when you run the next one, if you are not getting this characters in your specimen, then see whether the pseudobulbs pluriarticular or inflorescence with 1 to 2 flowers. If this is the condition, then go to the next one. You can see the pluriarticular leaf, I mean the pseudobulbs. Okay, then inflorescence with 1 to 2 flowers. There were no flowers for these bracts, and there were only flowers at these bracts, and you can see the flower, I mean the fruits here. It's very tiny. This is also about 2 millimeters, hardly 2 millimeters in size. You can see the drying flowers on top of the ovary. Okay, then yeah, it is area exilis. This one is area exilis, and if there are no inflorescence, it would be very difficult because it is very camouflaged to the substratum that it is growing. Okay, then if it is not this one, when you run the third key, then see whether leafless when in flowers, scape arising between the leaves, which is long and slender. Okay, and if this agrees, then go to 4. With 4a, internodes of the stem 2.5 centimeter or less. Then lip cuneately oblong, lobes obscurate, and cuneately oblong. This is, you can see that I mean towards the base it is little cuneate, narrowing down. Okay, then lobes obscure. You can, I mean, it's very difficult to see from the picture. Let me check whether I have now. Okay, so then if these conditions are agreeable, then it will be an area passiflora. And this, I mean, even without running the key, you may be able to identify, once you have identified this, because it do have a very peculiar character of
The speaker will be joining again. Kindly wait. Wait for some time. Dear participants, kindly wait. There is heavy rain in Kerala where the speaker is presently there. So he'll be joining us within 5 minutes or something.
Srujan, meanwhile, if you can give any suggestions to or instructions to the participants, please you give.
Madam, okay. I'm working on the other things.
Okay, okay.
Sujan, please check. Sir has joined, I think.
Sir, I'm continuously checking, sir.
Okay.
Sir, if you are there, kindly raise your hand, sir. It will be easy for me to find you out.
Dr. Zibi, sir, please raise your hand.
Sir, is this Sridevi, your profile name? I'm lowering the hands. Others don't raise their hands.
Yeah, dear participants, morning you have received the form. Please fill it carefully so that same name will be reflected on the certificate. Last time also someone not filled correctly, and it is again burden to our technical team members. So please be careful and fill all the columns carefully and send to the mail. Our technical team is overloading with continuous sessions, and tomorrow also we are going to start one more short-term course that is on green audit. So don't expect immediately your certificates. Surely you will receive the certificate short span of time. And one more thing, one more important thing is don't leave the groups until you receive the certificate. Once we dispatch all the certificates information to you, we only from our side, we close all the groups. Sir, have you joined? Dr. Amanchi?
Yeah.
Sir has some problem with the internet connection. He's trying to build, but unable. could you start that presentation?
I'll see 1 second.
Yeah, please.
Meanwhile, let me talk.
Yes.
Dear participants, so if you have wrongly filled your certificate form or something, you can edit that form. There is an option called edit. We have enabled that. So you can go back to the same, using the same link, find that edit option. If there are any mistakes, rectify them, correct them, and then you submit again, so that it will be good for us. Thank you. Meanwhile, I'll share you the feedback session link also after the Nageshwara's the valedictory session. So kindly do not expect the feedback from till that. After the valedictory section, we will be giving you the feedback from also. Those participants who have filled the contacts form, there is no necessary for messaging again. So your responses will be saved in the Google. So I'll be that will be reflected in the next PDF form. So don't worry about that. Over to Nageshwara, sir. Sir, unmute your mic, sir.
Is it okay, Srijan?
Yes, sir, it's fine, sir.
Yes.
Can I go ahead?
Yes, sir.
Yes, sir.
Yeah. Good afternoon, all the participants. Actually, we want to share this information with you during the entire 6 days course. But because of the tight schedule, we have not shared this. Because of the, even it is not exactly related to the plant sciences. But anyway, we want to, I want to share this with you. It's a kind of instrument, very user-friendly instrument, which was made available with the one of the scientists based US. Now, presently, this project is going on from the Department of Biotechnology, Government of India, Ministry of Science and Technology, in which I have also received a small grant. That is nothing other than the fold scope. It's a paper-based fold scope. It can be used. It can be made as a microscope. It is similar to the microscope. It can be used as a microscope in the field studies especially. And you can see in the picture that left side, this is Manu Prakash. Manu Prakash is a scientist, assistant professor in the Stanford University. And the right side picture also you can see, this is Dr. Jim. He is also a friend of the Manu Prakash. Manu Prakash is the Indian origin scientist in Stanford University, and Jim is the US native. And they are working together basically, and they have developed a microscope. What is the need of this kind of microscope in these days is, whenever you see microscope in any college, any school, any university, what we see is it is a very, very important one instrument, and it should be handled very carefully. Because of the extra care or personal care what we take, we don't allow other students, or we don't give the instruments to the students to handle it very freely. So that is a very much a limitation for the students to enjoy the science in these days. So that is the idea behind the developing of the fold scope in this project. Okay, so this is what a fold scope is. It is there in the first top center one, and that is blue color one. Yeah, you can see this is a fold scope. It can be assembled. We can assemble very easily that one. assemble it very easily. And what we need for the collection of the water samples, or what we need for the collection of the plant samples, or any material, whatever you want to do, some experiment, some kind of exploration, you can use these glasswares, scissors, very basically available in any stationery shop or any institution, slides, cover cavity blocks, a plaster, a cellophane tape, and small droppers, and this is the foldscope. Yeah, this is my students. They have collected water samples in different parts of water bodies with the waterlogged areas in Hyderabad, Usmania University, Hyderabad, and different parks also. They have collected water samples, and what is the aim of this study, or what is the use of the study, is to identify different kinds of zooplankton, especially ciliates and other larvae and other microalgae in this. I will not go through all the details because we have very little time. And anyway, I will share the details where the foldscope is available, how it is useful to the scientific community. And this is the sheet which comes along with the foldscope. And this is you can see in the top right, that is a foldscope compact box, geometry box like. The entire paper will come from that. And you can just simply, you can take out the paper and you can divide. There will be instruction guidelines for the attachment of the foldscope. It will be very easy, like it is a kind of toy. It is a kind of a paper-based toy. So simply detach the different parts of the foldscope paper, and then we have to attach, assemble again. And you can see here lenses, black color lenses will be there. And it will be, these lenses will have magnetic capacity, magnetism. So they will attach, you can attach these lenses. Once it is fixed with the foldscope, you can attach with the iPad, we can attach with the smartphone. So that is indirectly you are attaching a smartphone to the specimen, whatever specimen you want to see, so that you can take directly photographs. In a conventional microscope, we don't take any photos, just we observe the things. So because to avoid that, or you can to make it a user friendly, the foldscope has that provision. So that is a magnetic couplers. That magnetic couplers will attach to the we can attach to the phone, any smartphone. Okay, again, this is the ciliates introduction. It is not so needed. And we collected different water samples with the help of my students. You can see the pictures here. One picture left side. So this is Paramecium caudatum. So we have collected water samples, and we have taken a small drop of water, and it is fixed, and it is kept in the inside the foldscope, and observed the water sample. What are the different organisms are there in that? And we can see, we have taken all these photographs with using a regular smartphone. Any company smartphone can be useful. And here the first picture is Paramecium caudatum. There are so many organisms are there, and you can see we have seen divisions. Okay, in the binary fission or binary division in the asexual reproduction in the Paramecium caudatum, which is also recorded. I have got live videos with me, which are captured with the foldscope, and it is called, and third one is called stentor. It is again, it is a protozoan ciliate that can be also one of the good biological indicator of the water quality. And fourth one is Frantonia. Actually, this Frantonia, we have collected water sample from the Usmania University. Besides botany and zoology department, there is a waterlogged area in which we have identified Frantonia. It is not new, but it is there, but it is not available in other water samples. And third one is Spirostomum. It is also from the Usmania University water sample. It is very, very important specimen in the water quality. And once upon a time in the Hussain Sagar, there are plenty. When it was a freshwater body, means 30-40 years back, when it was a freshwater body, there are so many researchers have assessed the quality of water, and biologically and chemically at water quality also. This Spirostomum was there. There are once I have identified this Spirostomum, I have gone through the literature, and Usmania University itself has produced some of the papers in this Spirostomum. So it is there in the it is there in the Eastern Sagar water sample. And you can see the third next slide. That is next figure, that is verticella. Wherever you see water sample, a verticella campanula will be there. Actually, it is not a verticella separate one. It is attached with the, or it is there along with the cyclops. Yeah, next slide we'll see. So you can see tardigrade, figure number 7. It is very important zooplankton, tardigrade. It is a separate phyla actually, and tardigrade can survive any harsh condition up to 100 degrees centigrade. Wherever hot springs are there, this tardigrade will be there. Wherever cool water bodies are there, cool springs are there, that is in that also tardigrade will be there. So the tardigrade is an elephant, appears like an elephant. It can sustain any environment, and we have identified this tardigrade, the sample which was collected in the EPTRI institution. In the EPTRI institution also, when you go there, there will be a very big man-made pond at the center of the institution. So when I went there, I have collected water sample. When we observed, we have identified tardigrade there. And second one is figure 8 is cyclops. You can see cyclops very clearly, and in the conventional microscope it is very difficult to see. Because we'll take large amount of water sample, and it is they are very fast moving. Whereas in the foldscope, we'll take very little amount of water sample so that we can simply identify the organism very easily. And you can see the complete asexual reproduction, egg pouch of the cyclops egg case, and it is 4 o'clock plant stigma. It is also we collected in the Nizam College garden itself, where Vijay Bhaskar Reddy's are developed a very good medicinal plant garden in which we have collected the stigma of the 4 o'clock plant. Even along with this stigma, because I am from the zoology side, that is why I kept only these photographs. But we have identified, we have seen so many pollen grains as part of work in the foldscope grant. So you can anyone who is interested to identify pollen grains, to see the structure of the pollen grains, or to see the diversity of pollen grains, or even the other parts of the any plant, you can use the foldscope. Yeah, these are other pictures. Yeah, these are other pictures. Frontonia again. And this is Lobzodus. This is also Lobdosus species, which we have identified in I think in even Iswan University water sample. And Daphnia also, you can see Daphnia very much. And this is the larvae, Nauplius larvae of the Daphnia. We can see so many larvae. We have identified, we have seen mosquito larvae, so many mosquito larvae. We have seen dragonfly larvae. We have seen the venation in the different flies. I think I had those slides also there with me. Yeah, this is a Paramecium caudatum culture, complete culture. You can see the binary fission. You can see in the picture in the center of the center figure. In that you can see white circular kind of thing in the at the center of the paramecium. That is none other than the nucleus. So up to nucleus level you can see in the foldscope. And this is Euplotus, again the last picture, a Euplotus. It is binary fission of the Euplotus. It is also again a protozoan ciliate. Yeah, this is different kinds of abnormalities under stress condition in the paramecium. We have identified certain phytoplankton, phytoplanktons also, as I said. This is Chlamydomonas. You can see Volvox, Spirogyra, Actinastrum, Euglena, Pandurina. You can see Pandurina here. Spirulina, you can see. Gonium, you can see this is a star. 4 dots are there. It is very in picture. It is not so clear, but we have seen observed in the foldscope. It is very clear. It's a Gonium species. So like that, if anybody interested, you can see different kinds of algae in freshwater bodies with the foldscope. Yeah, this is another kind of study we have done with the foldscope. This is collection of the collection and identification of the different venation in different insects. You see here cockroach. We have identified. We have collected the cockroach wings and identified how the venation is, how the color is, because it depends. The color depends on the different animals. So how the color is, how the venation is, how the circulatory system will be in different wings of the insects. It is also a small task I have given one of my student, and he collected a cockroach wing. You can see the pictures. 2 pictures also the same. Yeah, this is housefly wing. You can see very hairy kind of thing in here, hairy kind of extensions at the margins of the wings of the housefly. See, venation, it is very much simply clearly different from the cockroach. Dragonfly, again venation in the dragonfly, wings of the dragonfly. So it is completely different. And coming to the last picture, that is a butterfly. So you can see these so many scales on the wing of the butterfly, and the scales are pigmented. Sometimes along with the scales, the other part of the wing also pigmented. So the color of the butterflies, because they are in different colors, the color of the butterflies comes from the scales which are pigmented in different colors, and in different area of the each and every wing also color is different. So you can identify coloration, you can identify pigmentation, what kind of pigmentation, and you can identify venation in different wings with the foldscope. Yeah, I have organized outreach programs on 26th September 2019, and also in different colleges and institutions. How this foldscope is useful. It is organized in the CCMB Open Day, and the CSIR all institutions Open Day is on 26 September. It is celebrated like that. And we had a foldscope a stall. During the stall, so many students have come, and we have given the demos there and then. Yeah. With this small presentation, I thank all the participants. And coming to the, if you're really interested, you can go to the details. There are just in the net, you go to the internet, any internet, Google, and just you type foldscope, and foldscope website will be visible. And that foldscope using that foldscope website, you can purchase your own foldscope, which is available in the Flipkart, Amazon, or you can from the foldscope website also directly you can purchase, and it is costing around 500 rupees. So it is very low cost. You can easily handle, you can carry to anywhere. And field studies, especially field studies, any botanist who is interested to identify any kind of sample there and then to see, to observe, then you can carry this foldscope. So it is very user friendly, and it is very low cost also. With this, I'll stop here. Yeah. Yes. So now this Manu Prakash is coming with another kind of centrifuge. It is also paper kind of centrifuge. They are coming with different centrifuges, and they are coming with other different kinds of small kits kind of. So small toy kind of scientific instruments. They are very user friendly, handy also. And thank you for the opportunity, Dr. Vijay Bhaskar Reddy.
Sir, great, sir. Really, this foldscope is too handy. Very great. You also answered the basic question, sir, because some people asked about the cost and where it is available, both in YouTube and here.
Yeah,
you have already made the point, sir. Thank you, sir.
Thank you. Thank you. Thank you, Srijan. Actually, I have so many other pictures and other details, but it is now we have very limited time. I have made this presentation, I made it ready. If any technical snack comes, to present to the audience. Thank you. Thank you.
Thank you, sir. We have failed to get back on Gibbs, sir. Sir is not available due to heavy rains in Kerala. So over to
then I request Dr. Amanji Nageshwara to propose a vote of thanks to the Dr. Zibi.
Yeah, thank you, Vijay Bhaskar. From the on behalf of the organizing committee and on behalf of the short-term course on plant taxonomy, we are very much thankful to the Dr. Gibi. And actually, we have missed, as I said in the beginning during introduction, definitely it will be a very good presentation because we can see so many kinds of orchids. Actually, I have a doubt in my mind to ask our resource person, because nowadays wherever you go outside, we will find so many kinds of plants, so many kind of ornamental plants. But these ornamental plants are all imported. They are not native of India. They have imported from US, Europe, so many other countries, not native. Very few are native. A majority are outside. So is there any mechanism, or is there any efforts from the government of India to see that when different kinds of orchids of local varieties are can be propagated, can be maintained, can be made a common orchid in the orchids are ornamental kind of thing in India. But anyway, because of the technical snag, that resource person is not available, not connecting to us again. On behalf of the organized committee, I'm very much thankful to you, and we will be reaching you through mail, sir. And thank you, Vijay Bhaskar. Now I request Sadhana Srijan to continue.
Yes, sir.
Sir, I think Vijay Bhaskar Reddy, sir, I need to reflect on this again. Sir, unmute your mic, sir. Nageswar, sir.
Sir, Vijay Bhaskar, sir, please unmute yourself. Anyway, any other instructions? So
Sir, can we start the valedictory section, sir?
Yeah. Professor Satyaprasad is there in the list, you see, please. Professor Satyaprasad. Professor K. Satyaprasad.
Satyaveni. No, sir, Satyaprasad sir is not available. Sir, if you are there, please raise your hand, sir. Or,
Professor K. Professor Kata Satyaprasad. And one more, Mathur also. Vinod Mathur, you find out, Srijan, please. Anyway, 3:30 now, we said 3:30, they will be joining mostly.
sir, couldn't find them, sir.
Yeah, no. VBN, sir is joined just now, it seems. You find out. VBN. Vinod B. Mathur. Sir sent a chat box message. VBN.
Sir, can you take Sharin, sir? From my side, there is a glitch.
VBN. Yeah, I'll see.
Yeah, good afternoon. I'm Dr. Mathur here.
Yeah, sir. Yeah, thank you, sir.
Can you hear me, see me?
Yes, sir. Yes, sir. We can see, sir. Thank you, sir.
Okay.
Welcome, sir.
Sir, welcome, sir.
Yeah, good afternoon.
Great to see you.
Good afternoon, sir.
Sir, few minutes, sir.
Sure.
Sir, Dr. Avinash Jaiswal, sir, welcome, sir. Sir, please turn on your video, sir. Welcome, sir.
Sir, good afternoon, sir. From Sir Avinash, sir, Vice Principal, Nizam College. This is Nageshwara, sir. Sir, unmute your mic, sir.
Hello.
Sir, audible, sir. Now you are audible, sir.
Thank you.
Yeah.
Thank you. Thank you.
Welcome, sir. Welcome, sir.
Thank you very much. Thank you very much, Nageshwara. Good to see you after a
Thank
long
you.
time.
Yes, sir.
Sir, namaste, sir. Welcome, sir.
Thank you. Thank you very much, madam.
Thank you, sir.
Shashikala madam, there is disturbance from your mic, madam.
What should I do now?
Kindly mute your mic, madam.
Srijan, is it okay now?
Yeah, over to Sadhana.
Madam, noise from your phone, madam.
Now also it is noise.
Yes, madam.
Yeah, Sadhana, please.
Yes, sir. Good afternoon to everyone. I welcome Dr. Mathur sir, Dr. Avinash Jaiswal sir, and all the organizers to the valedictory session. As we come to the end of the national level short-term course by Department of Botany, Nizam College on plant taxonomy, allow me to quote Dr. E.K. Janki Ammal, the first woman botanist in India, and was one of the pioneering force of botanical sciences in the post-independent years of the emerging India. As she says, my work is what will survive. I will be remembered by my work. And hereby remembering the legendary woman, Dr. E.K. Janki Ammal, for all her contributions to the botanical sciences, we start the valedictory program, and I hereby invite Dr. D. Seshikala, Assistant Professor, Department of Environmental Science, Osmania University, to give away the welcome address. Over to Seshikala, ma'am.
Thank you, Sarna. Thank you very much. Is my voice is clear now?
It's clear, ma'am. Please carry on.
Okay.
It is clear, ma'am.
Yes, thank you. Thank you. Good evening, all. Now we have come to the last part of our journey. This short term was short journey, but it was very interesting and very enthusiastic, and we all empowered with full of knowledge, especially in plant taxonomy. To this August gathering, the valedictory function, I invite Dr. V.B. Mathur, Chairman, National Biodiversity Board. Sir, please you join with us, sir. Invite our today's guest of honor, Dr. Avinash Jaiswal, Vice Principal, Nizam College, Osmania University. Sir, please, I welcome you, sir.
Thank you. Thank you.
Next, thank you, sir. Next, I'll invite another guest of honor, Professor K. Satyaprasad, sir, a retired professor from Department of Botany, University College of Sciences, Osmania University. I welcome you, sir. Now I'll invite Dr. Vijay Kumar Garu, Academic Coordinator, Department of Chemistry, Nizam College, Osmania University. I welcome you, sir. So I welcome all the participants to this August gathering. Now we have come to the ending of this short-term course. At this juncture, I would like to tell a small short story regarding this short-term course. So earlier in the on the date from 8th to 9th, we conducted one international conference. So by seeing the response from these different participants, we got good strength, our motivation power to run even this course also. So this plant taxonomy was a specialized area, and that too it is less given importance. So that's why we thought of organizing a short-term course to have proper knowledge among the participants. So in our effort, we could get all the different distinguished eminent speakers to deal with different subjects, and our motive was to spread this knowledge to the across the country, and to our motivation. Even in this pandemic situation also, the participation was encouraged. Participation was encouraged with nearly 300 live participants, and maybe 100 or more in YouTube participation. So this gave us still more strength, and till now, today, at this juncture also, I think nearly 170 participants are seen here, it seems. We are very, very happy to have you all here. I think you are overwhelmed with the knowledge, especially in this plant taxonomy and all other sub areas which were there, which were dealt by different speakers. So with this small note, with all the encouragement from the speakers as well as our higher officials, and also from our technical team and our organizers, we all are very, very thankful to the participants also. Thank you very much for giving me this opportunity. Thank you one and all.
Thank you so much, ma'am. Now I invite Dr. A. Vijay Reddy, sir, Assistant Professor, Department of Botany, Nizam College, to give away the short-term course report. Over to Vijay Reddy, sir.
Yeah, thank you, Sadhana. Am I audible?
Yes.
Yes, sir. Yes, sir, you're audible.
Yes,
respected Chief Guest of the Day, Dr. Vinod Madhur, Chairman, National Biodiversity Board. Today's special guest, Professor K. Satyaprasad, and today's guest of honor, Professor Avinash Jaishwal, Vice Principal, Nizam College, Osmania University, Dr. Vijay Kumar, Academic Coordinator, Nizam College, other dignitaries, organizing committee members, their colleagues, and participants from all over India and abroad. A very good evening to all. 1 week national short-term course on plant taxonomy organized by the Department of Botany, Nizam College, Osmania University, Hyderabad from 3rd to 8th August 2020. The inaugural session began with the welcome speech delivered by Professor Lakshmikant Rathod, Principal, Nizam College. Later, Chief Guest address was delivered by Professor C.H. Gopal Reddy, Registrar, Osmania University. We got a good appreciation and encouragement from our Registrar as well our Principal, Lakshmikant Rathod. That make better us to organize this course and ended very successfully. The first day was commenced with the outstanding presentations by the Dr. Sanjappa from Bangalore, Professor Pullaya from Andhra Pradesh, and Professor Vatsavya S. Raju from Warangal. Day was indeed very informative as well as enlightening. Then it was succeeded by the astonishing demonstrations by Dr. Karupaswamy from Tamil Nadu, Dr. Sudhakar Reddy, NRSC from Hyderabad, Dr. Mayur Nandikar, Maharashtra, and Karnakar Reddy from Hyderabad on the second day. These presentations were very remarkable in their own way. The next day anxiously arose with the spectacular illustrations by Dr. Narsimhan from Tamil Nadu. Whole session, morning session was dedicated by the Dr. Narsimhan, a very elaborative session on the classification systems in general and APJ classification in particular. Dr. Mayur Nandikar and Dr. Swami BSI Hyderabad, who also resolved all the queries of our participants, which had with a lot of endurance. Subsequently, the fourth day surfaced by an astounding presentations of Dr. Vijay Shankar Raman from USA, Dr. Karpaswamy from Tamil Nadu, Dr. Ramakrishna, and Dr. Upender from Osmania University, Hyderabad. These were exceptionally impressive to our participants. The next day focused entirely on the illustrations of Professor Ravichandran from Tamil Nadu, Professor Kashinath Bhattacharya from West Bengal, Dr. Sridvas from Osmania University, Hyderabad, and Dr. Madhusudan Reddy from Kadapa, Andhra Pradesh. The participants were very much impressed as they were very much satisfied with the wonderful information and delivery of each presentation. Ultimately, on the day of conclusion, that's today, ushered with the eye-catching presentations on the types of taxonomy brought out by Professor A. Nagamani, Osmania University, Dr. Mamata Bhatt from Jammu Kashmir, Dr. Zivi Kuryakozi from Kerala. Because of the power cut, we couldn't see the whole presentation, but it was an outstanding presentation. It was in fact very tremendous. Each and every one of our resource person accepted their extraordinary efforts, which has hopefully enriched the knowledge of our eager participants day by day. I extend my deep gratitude to everyone who made this 1 week short-term course a successful one. I sincerely thank our registrar, Professor C.H. Gopal Reddy, our beloved principal, Professor Lakshmikant Rathod, Vice Principal Dr. Avinash Jaiswal for their constant support and encouragement. I also thank our co-organizing members, Dr. Shashikala, Dr. Nageshwar Amanchi, and back-end technical team, Srujan, Sadhana, and Karthik for their strong vertebrate support. At last, I appreciate all our participants who spend their valuable time, interest, and their involvement in seeking more wisdom about plant taxonomy. Stay home, stay safe, stay knowledgeable. Thank you one and all. Over to Sadhana.
Thank you, sir. Hereby, we would have the feedback from the participants, sir.
Yes, Srijana.
People who wish to give the feedbacks, please raise your hand so that we could add you into the panel. Or if there are any people listed out earlier, Srujan, would you please make us aware of who are going to give their feedbacks?
Yeah, people are not available. I am checking them. But Jay Swami
Your voice is very low.
Jay Swami is there. Let us ask him to respond.
Jay Swami. Sir, please give away your feedback by unmuting your mic. Jay Swami, sir. Jay Swami.
Dr. Parmesha.
No, he is not yet added to the panel.
We'll take Dr. Akbar Basha.
Sir, good afternoon, sir. Am I audible, sir?
Yes,
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 sir.
yes, sir. Thank you.
Sir, good afternoon, sir. Basically, I'm a plant physiologist. I did my research in plant physiology. Previous days, I feel that taxonomy may be a kind of endangered subject in the sense that due to lack of knowledge or some other means. But after attending this short-term course of 6 days, really I recognize the potential, how it has incorporated into so many areas like in molecular plant physiology, molecular biology, and in some of the aspects like in APG classification. We really wonder how the DNA and other biochemicals and other chemicals play a vital role in understanding the plant physiology, plant taxonomy. Then later, I am right now I am in a position that no subject is endangered. Every subject is growing in its own way. This was my false conception in my mind. Thank you, sir, for arranging this wonderful session, sir. Thank you, sir.
Thank you so much, sir. Next, we would go with Dr. Paramesha Malligauda. Please unmute your mic and give away your feedback, sir.
Thank you, ma'am. Good evening, all. I am Dr. Paramesh, Assistant Professor, Department of Botany, St. Joseph's College, Bangalore. Though COVID-19 has stopped many things in the world, but not for teaching and learning. First of all, I congratulate all the organizing committee members, especially Dr. Vijay Bhaskar Reddy sir, Dr. Amanchi Nageshwar sir, Dr. Sasikala ma'am, Sadhana ma'am, Sujan sir, and Karthik sir for organizing such an intensive course. It's very neat and well organized. And in fact, the topics were also much diversified from morphology to molecular level, which covers almost all aspects of botanical research. Many topics was quite challenging for resource person to communicate to participant, but our resource persons actually made the topics very easy, interesting, and simple manner. Okay, so in fact, to get that 1 hour knowledge, at least we have to read more than 10 books and hundreds of research paper, but we got those knowledge in 1 hour. Okay, so we are very fortunate to have such eminent and renowned speakers. So thank you all, dear sirs and ma'ams. And again, I thank all organizing committee members, especially Dr. Vijay Bhaskar Reddy sir, to organize such few more courses in the near future. We are very happy to join. So thank you all. So may God bless us all.
Thank you so much, sir. Next, I would request Mr. Bidan to please give away the feedback. Sheikh Makbul, sir, are you there?
Yeah, madam. Yeah.
Yes, sir. Please give away your feedback, sir.
Yeah. Thank you very much to the entire crew of Botany Department of Nizam College for organizing this wonderful event on plant taxonomy. As we all botanists know that plants are becoming endangered, even animals have become endangered or extinct. And even if we see nowadays, people say that humanity is also being endangered. As we have seen that, especially in plant taxonomy, very few people are working. So due to this, as earlier speaker said, due to this, the taxonomists are also becoming endangered. So to keep this subject in life or in track, I thank the organizers for taking pain in organizing this event with very relevant, very relevant and very important topics uh, uh, from classification to molecular level. All the areas which was covered during the last 6 days by our resource persons was heart-touching. I appreciate Dr. Vijay Bhaskar Reddy is a very close friend of mine. Dr. Vijay Bhaskar Reddy, Dr. Nageshwar Rao Amanchi, Dr. Shashikala, for maintaining even timings of all the session very promptly. I have never seen in any kind of any kind of webinar, nor even in the live seminar, that these people have starting session exactly at 10 o'clock means 10 o'clock, and say, and even afternoon 2 o'clock means exactly they were starting at 2 o'clock. I appreciate really. And although we were not having these sessions and lunch sessions in this COVID pandemic condition, but we all had virtual lunch in all the 6 days. So finally my sincere gratitude to entire team of teaching staff of Nizam College, as well as technical team, Srujan, Karthik, and also to Sadhana. Thank you very much, sir. Thank you. Thanks.
Thank you so much, sir. Any other people who are present, please unmute your mic and speak.
Betsy is there.
Betsy, yeah, please give away your feedback.
Warm and graceful to our most valued honorable chief guest, organizing committee, and all my participants. It's my privilege to give the feedback of this course and acknowledge of those who worked really hard to make this course happen. I'm Betsy M. Koshi, a third-year BSc student from Nehru Arts and Science College, Coimbatore. I would like to thank all the 18 expert resource persons who gave their 200% to enhance our knowledge. I could find that all of them were so excited and honest of taking classes that they could take classes continuously for 2 hours without even taking a break. Next, I would like to thank the heart and soul of this course, the organizing committee, Dr. Nageshwara Rao Amanchi sir, Dr. Vijay Bhaskar Reddy sir, Dr. Shashikala ma'am, Sujan sir, Sadhana ma'am, and Karthik sir for working tirelessly and making this course a huge success. My heartfelt thanks to Nizam College for encouraging and enlightening the youth of the nation about the needs of this endangered subject, plant taxonomy. I really would like to thank Vijay Bhaskar sir, especially because I am a BSc student, and when I joined this course, he told me that this course is not for BSc students, it's only for PhD, PG and above. And I requested him to accommodate me. I would really thank him, because if he wouldn't have accommodated me, I would have never known about plant taxonomy in depth like this, because I'm from biotechnology background. So according to my knowledge, I know only Carl Linnaeus binomial nomenclature that I studied in 12th. From there, the technology, the history of classification came to APG classification. I never knew that. And telling about phonetics, cladistics, phylogeny, everything was very new to me, and I'm very thankful to all the organizing committee and the university. Thank you. Thanks.
Thank you so much, ma'am. I would request Dr. S. Datta to please give away your feedback, sir.
Good evening, all. I am Dr. S. Datta from Rajasthan University, Jaipur. It has been a very, very enlightening experience for me, and I feel we have to take this forward. Nizam College has done a wonderful work. They have introduced these new resource persons to us also in North India, especially in Rajasthan, where the ecosystem is very different from yours. And I'm going to carry forward these lectures, these examples, some new things which I have also learned to my students, and try to collaborate with some of the resource persons you have introduced to take these lessons forward. We must just not listen to the lectures and, just sit on it. There has to be some action-oriented work also, and I'm thankful to you all for giving me this chance to come up with some ideas and introduce that in my college. Thank you all. Thank you very much.
Madam, thank you very much, because this was the main motto of conducting such program, ma'am. Thank you very much that you have you told that this knowledge will be spread to your students, as well as you are going to have collaboration even with the resource persons, and also
Yes,
the
ma'am. Also
different.
with your college.
Yeah. Thank you very much, sir. Thank you
Thank you,
very
ma'am.
much, ma'am. Thank you.
Thank you so much, ma'am. I would request Maheshwari to please give away your feedback, ma'am.
Hello, ma'am. Am I audible, ma'am?
Yes, ma'am, you're audible.
A very good afternoon to all. I would like to thank all the organizers for organizing this wonderful 1 week program e-certificate course. And I'm very happy that I got a chance to attend this 1 week program. The very first day when I got the brochure from some forwarded message in WhatsApp, the very first day I gave 1 week leave for my duties and got ready to attend these all sessions. And I'm very happy that all the sessions were very fruitful, and especially the topics highlighted upon the Commelinaceae, Poaceae were very informative, and I hope that from these sessions we got the maximum information regarding the taxonomical information and identification of the plants. And I would like to thank all the organizers from all the participant side and my side that they conducted this wonderful program, sir. Thank you, ma'am.
Thank you so much, ma'am. Srijana, are there anyone else to speak? Yeah, I request Megha Shri to please give away your feedback, ma'am.
Is I audible, ma'am?
Yes, ma'am, you're audible. Please a bit louder.
Okay. Good afternoon, all. And first of all, I thank all the organizers to provide a platform to learn about taxonomy. And I am very grateful to you all, because as a student of taxonomy, there is no end for learning. And I got much and more information about the family Commelinaceae, where I am mostly I am much interested on that family, because the flowers are very attractive, and I got very much information about that. And the resource persons from different background, like molecular taxonomy, cytotaxonomy, and these are very informative. And once again, I thank all of you for providing this opportunity.
Thank you so much, ma'am.
Sadhana, 1 minute.
Yes, ma'am.
Satyaprasad sir is on to panelist, it seems. Satyaprasad sir?
Yes, Satyaprasad sir has been added to the panelist, ma'am. Sir is in the panel.
Okay. Sir, we welcome you to this August gathering, sir. Thank you very much, sir, for your presence, sir. Thank you very much, sir. Yes. You can continue.
Are there anyone else to speak?
Yeah, this is it, Amma. We can go with the next.
Sorry, who?
I think we can continue. No, there are other participants raising, but I think.
Yes, we'll move to the next part.
We shall move.
Okay. Now going on with the valedictory session, I invite Dr. Vijay Kumar, Department of Chemistry, Nizam College, to give away the address by academic academic coordinator address.
Sir has not joined.
Sorry, sir?
Sir has not joined yet because some technical issues.
Shall we go on with Satya Prasad sir?
Yes.
Yeah. I welcome Satya Prasad sir, our guest of honor, and I invite sir, Professor K. Satya Prasad, Professor Retired, Department of Botany, Osmania University, to please give away his valuable address to the session.
Sir, kindly unmute your mic, sir. Satya Prasad sir. Unmute your mic, sir. You can find the option on the left down corner.
Okay.
Yes, sir. Thank you, sir.
Yes, sir. You're
So, good
audible.
afternoon, everyone.
Good afternoon, sir.
At the outset, let me thank the organizers, particularly Dr. Vijay Bhaskar Reddy, for making me once again a member of the family of botany or the plant scientists. So, having retired from the service, in fact, I'm not really into the subject or participating in any of these things, but you have made it, particularly in the time of pandemic, you have made use of the time, and you have having heard about many feedback and people participants. I understand that really it is a national symposium or seminar, whatever you may call. There are very many interested people from different states of the country have participated. And if you want to conduct such a seminar, in real terms, it would be highly difficult. But you have made use of this opportunity, and as it is said, only successful people can make the opportunities as the vehicle of success for them. Hope this would help all of you, Dr. Vijay Bhaskar Reddy, Dr. Shashikala, Dr. Nageshwar Rao, Dr. Spandana, and many of my friends sitting there participating in this one. Coming to the topic taxonomy, people say that taxonomy is the mother of all sciences. Having said that taxonomy is the mother of all sciences, the problem is also there because in real terms, the mother and father in day-to-day life are in the old age homes. Similarly, the subject taxonomy is also highly neglected. Rather, the so-called modern subjects have come and they have seen that the taxonomy is nothing and it has no importance and so on. And ultimately in the universities, maybe for different reasons, taxonomy and taxonomists have been neglected. Unfortunately, we have reached to a stage that we are not able to identify, or in a coming days probably, we may not be in a position to identify a species, maybe a plant species or animal species, or for that matter, a microorganism. So a situation may come one day that we may have to import people from outside. I understand, and also we have the practice in plant sciences for taxonomy, we follow the Gamble flora of the Gamble flora. Still, I think we are following, and a wonderful thing he did, and then there is no comparison with that. But still, probably one more Gamble we may have to invite, or rather make him come back to our country in order to identify our plants. So identification, one may ask, there is a famous Shakespearean saying that what is there in a name, but there is a lot in its name as well as in the way how you place it and where you place it, how you understand it, and particularly in these days, plant genetic resources have become one of the important resources that are being exploited commercially, and they are being patented, and they are making use of their potential in the business, whatever. So in that sense, if you are not able to identify, you are not able to classify your own resources, maybe plants, animal, or whatever, microorganisms, all these resources have to be, your own resources have to be identified and listed, and that has become neglected. And in that sense, what happened, we have seen the examples of turmeric, we have seen the examples of basmati and so on. So people claim that those particular ones which belongs to us have been claimed by many others, and again we have to enter into the litigation and say we have to prove ourselves that turmeric is ours and so on. So by opting or rather studying is not just not just for livelihood's sake. Most of our friends are their associate professors, assistant professors, and so on. So maybe that may be helping them as a livelihood, but at the same time, they are doing a great job. It is like a what do you say, a patriotic job. If you are really doing taxonomy, you are doing not only for your own personal end, but for the purpose of this, for the benefit of the country. So benefit of the society. So what is request all the participants, take the spirit of taxonomy, and this particular taxonomy is going to help not only yourself, but also the society. So this particular thing needs to be ingested into the minds of our students. Unfortunately, the basic sciences like botany are generally they are considered as food courses. My university friends very well know it. But that particular food course is going to help us to get food security, and so on. So therefore, taking this particular topic of plant taxonomy and arranging a webinar in this particular pandemic conditions is a wonderful job you did. And the resource persons also, as I understand, they have been from various parts of this country, and they are very experienced, and they are authority in their own field. So to bring such people onto the platform is not a easy job. So your efforts should go in a right manner. Your effort should be fruitful, and particularly the subject of taxonomy, though we are dealing here in the webinar, but actually we have to take these things into the field, and the training conducting forays, conducting field trips, identifying the plants, and we should make the student to understand the importance of the biodiversity. So plant biodiversity, animal biodiversity, and what is the importance, what is the value of it, and what value it is going to tell to the society. So in that sense, my friends in the Uthmania University, I am very proud of you. You are doing a wonderful job and continuing the legacy. I remember my teachers at this occasion. So they used to feel a lot. There are people are becoming great plant breeders, or say the molecular biologists and so on and so forth, without knowing what is a flower, what is a female flower and what is a male flower. So that's what the situation has come. So therefore, we have to make taxonomy as one of the important aspects of science, or aspects of botany or zoology, and so therefore we would be able to give a comprehensive understanding of the subject. It is not compartmentalizing. Unfortunately, compartmentalization of the subject, maybe because of personal interests, personal things, we have already done a lot of damage to the topic, the subject. Therefore, it is time that we realize our mistakes and really study this particular taxonomy with the real thing. And the other parts of the country, of course, there are certain universities doing going into the science very seriously, and particularly in the state universities. So this particular subject is becoming lagging behind. So therefore, you take that for taking this interest and also the efforts you have put in to become fruitful. I wish you all the best, all the participants throughout the country. I wish them all the best, and still you make the flag of taxonomy very great in the coming days to come. So I once again thank you for giving me an opportunity to interact with you, and thank you very much. All the best.
Thank you very much, sir. You have credited us that we have again added you to the family, but you people are like the head of the departments, head of this thing, you people have initiated it, and you are the head of the family, so you people can never be out of the family. You are the one who guide the thing, all the other people in the family. So please keep guiding us, and I really thank you for whatever you've shared today, sir. Thank you so much for giving your valuable time. And now I would invite our guest of honor, Dr. Avinash Jaiswal, sir, Vice Principal, Nizam College, Osmania University, to please address the session.
Thank you very much, madam. Good evening, everyone. Respected Vijay Bhaskar Reddy, Head Department of Botany, Nizam College, Dr. Ramanchi Nageshwara, Department of Zoology, Professor Shashikala Madam, Environmental Science. So I can find three big departments in this one seminar. So this is like all in one. One is zoology, one is botany, and one is environmental science. And just lacking Mr. Vijay Kumar, who is from chemistry department. The four pillars are there. I am very happy and feeling very honored to be a part of this 1 week national workers seminar or workshop on this plant taxonomy. And on behalf of Nizam College, I congratulate one and all who made this program successful, directly or indirectly. And whenever any department or teachers require our services or our help or our cooperation, will be there for everything. And once again, I want to congratulate especially Dr. Vijay Bhaskar Reddy garu, Dr. Ramanchi Nageshwara, and Professor Shashikala Madam. Thank you very much for giving me this opportunity, because I have to attend one more webinar on English department. They are waiting for me. I'm very sorry to cut short my speech. Thank you very much and congratulations. And I would like to share this, that some persons told me that Vijay Bhaskar Reddy, we are having the plants which are having to cure the coronavirus. You know, we are having in this Nizam College Botany Department is having that cure in Nizam College. So we are also looking for that one. So whenever you feel free, Vijay Bhaskar Reddy, please come and contact me. You will find it out. Thank you very much, Vijay Bhaskar Reddy, and congratulations once and all. Thank you. Thank you very much.
Thank you so much, sir.
Thank you very much, sir. Thank you.
Yes, sir. Thank you, sir.
Thank you.
Thank you, sir.
Now I would hereby invite our chief guest, Dr. V.B. Mathur, Chairman, National Biodiversity Authority. After serving for 33 years at the prestigious Wildlife Institute of India, including as the Dean and as the Director, Dr. Vinod B. Mathur was appointed by the Government of India as full-time Chairperson of the National Biodiversity Authority in India in September 2019. He obtained his master's degree in zoology from the University of Rajasthan and doctorate degree from the University of Oxford. He joined the Indian Forest Services in 1983 and topped in the in-service professional training course at Indira Gandhi National Forest Academy, Dehradun, and also in the Lal Bahadur Shastri Academy of Administration in Mussoorie. Dr. Mathur has been actively contributing on research policy, interface, and biodiversity mainstream issues. He is currently the UN CBD Bureau member and UN IPBES Bureau member, representing the Asia Pacific region. Sir, it's our pleasure to have you here on this auspicious day, and I would hereby request you to give away your speech for the valedictory session.
Sir, kindly unmute your mic, sir.
Yeah, good afternoon, and to everyone. We have with us the principal, the vice principal, the course coordinators, as I heard them, and a large number of resource persons. But more importantly, the 150 plus participants that you have enrolled for this course from all over. I welcome you all. It's very privilege and both pleasure for me to be delivering this valedictory address on a thematic area that forms the core of the principles and practices of conservation science. It may appear to be an old discipline, but as our professor just said, it forms the core critical mass of all sciences that we deal with. This week-long course, I very interestedly look at the content, where you got the resource persons from all over and the presentations and the lectures that were made. I am very sure that a new sparkle has been put in this 250-year-old discipline. Now, this course is also very timely. We are no hesitation in saying that we are currently witnessing. Yeah. Can you hear me?
Yes, sir. Yes, sir.
Yeah, so we are currently witnessing a kind of a stalemate in field research with a lot of slowdown and restrictions that they are appearing. And therefore, it is very important to concentrate on the transmission of latest developments in science. And even a face-to-face course is difficult, but you people have shown that what virtual teaching can do. Now, undoubtedly, the plant taxonomy is the most basic and fundamental subject, and no other studies related to any aspects of botany, whether pure or applied, can be either complete or perfect. Careful taxonomic enumeration of countries' biota and understanding of the biological richness is a very critical requirement. Now, see, at the global level, what is happening, we call it biodiversity census, we call it counting of species. But this is very, very important, especially when we say that our country is one of the 17 mega biodiverse countries in the world. So how do we say that? How do we look at the status if we are not able to do a taxonomic enumeration of species? So it is this taxonomy which is the tool and lens through which we look at the biological diversity of Earth. The basic discipline, it gains more important because we know that the world is undergoing what we call as a biodiversity crisis. Taxonomy, therefore, underpins our ability to conserve and benefit from biodiversity in many ways. In fact, we have, as you would know, this big convention on biological diversity, which came into being way back in 1992. So this convention recognized that there was what I would call as a taxonomic impediment to the sound management, to the sustainable use, and to the conservation of biological diversity. And therefore, CBD recognized that there was an importance of pushing the science of taxonomy. And therefore, way back in 1995, the Global Taxonomy Initiative was launched. And governments were asked to submit a review, study what the taxonomic needs of the country were. And then CBD urged all countries to formulate what we call as a national program in taxonomy. Now, what we call as taxonomy Our modern day scientists and colleagues are saying it bioinformatics. There has been a revolution in information technology, but the taxonomic information has practically remained unchallenged for the last 250 years, ever since the famous scientist or botanist Linnaeus introduced the binary system of classification of living organisms. So we now have moved a huge distance. In the past 250 years, taxonomists have named about 1.78 million species of plants and animals and microorganisms. Yet, the total number of species which is unknown to science still ranges between 5 and 30 million. See the gap that I'm saying, 5 and 30 million, the range is very, very huge. So whatever unique ecological, genetic or biochemical properties these species possess, there is a possibility that we might lose them before we know them. And that is the reason why courses like this have a very important connotation. The science of biodiversity is underpinned by the advances in taxonomy. We do need to change our approaches for being a conservation-centric approach to a community-centric approach, from a logic-based approach to a legal-based approach, and from a policy-based approach to a practice-based approach. And therefore, I would urge the taxonomic fraternity to spend some more time in assessing the impacts of these approaches. You recall that in 1992, we had the Earth Summit. We also had established a fact that there are sovereign rights of the countries over their biological resources. And therefore, we must put in place a governance mechanism which takes place and puts a rightful place for the science of taxonomy. We are interestingly struggling to find the value of nature. We need to find out that what is the economic value of this nature that we want to protect and conserve. And sometimes this lack of valuation causes a kind of a degradation and loss of biodiversity also. So this is where we need to look at. And I will go to the extent to say that we cannot manage what we cannot measure. And therefore, our scientists, ecologists, economists are doing a series of assessments so that we are able to measure the value of this biological diversity and the science of taxonomy, which forms the basis of it. We have a Biological Diversity Act, and with me sitting in Chennai, which is the headquarters of the National Biodiversity Authority, we have a mandate to prepare what we call as a People's Biodiversity Register. So this is the document which contains information about our biological resources, about the medicinal values of the plant resources that we have. And it is in this context I would like to appeal to all the students, all the teachers, all the resource persons who are here, that we now need to move ahead. We now need to make our registers, the People's Biodiversity Register, more substantial, more comprehensive in nature. And therefore, this poses a great amount of taxonomic challenge to us, which the speakers before me, and I have tried to provide a perspective on it. And I would urge all the participants to carry forward their efforts. And these courses, they do a lot amount of networking. As I heard during the feedback, there is a lot you can learn. And the 150 plus people who are listening to this program and who are part of this very valuable course should not lose each other. And at any point of time that you need help from each other, from your teachers, from your mentors, please make sure that you approach them. I'm sure in this modern IT world, everybody would use an email or a WhatsApp to get to the other person. And I, on behalf of the National Biodiversity Authority, would like to assure you that any help that you as an individual or as a teacher or a practitioner of taxonomy would like to need, we would be, and my authority would be very willing to help that. So with these words, I thank you once again for providing me this opportunity. In fact, last week only when Dr. Reddy approached me that there is a program, I just checked with him and he said, yes, it is also being run on a Saturday. And I told him that then it will be a lot easy for me, because during the weekdays, I do have to engage in many other things. But today, I was available and I was very, very pleased to be invited and to address you. And I'm sure my words will carry some meaning in the years of the young students. And this discipline of science, whether it is botany or chemistry or pure sciences, and the science of taxonomy, we would definitely have to move towards molecular taxonomy, but the basic taxonomy will always be central to all the endeavor that we do. So with these words, I thank you once again, and I wish you all the best for your future career. Thank you once again. Thank you.
Thank you. Thank you very much, sir.
Over to you, Dr. Sadhana.
Thank you, sir. Thank you so much.
Thank you. Thank you so much, sir.
Thank you, sir.
Now I would invite Dr. Nageshwara Amanchi, sir, Department of Zoology, Nizam College, Osmania University, to please give away the vote of thanks for the session.
Yeah, good evening, all the participants, my dear colleagues, respected guests, Mathur sir, Professor Satya Prasad, Dr. Gibi, speaker in the afternoon session, Dr. Vijay Kumar, Dr. Avinash Jaishwal, Vice Principal of the Nizam College. Very good evening to all of you. We have arranged, as one of the participants said, a multiple number of resource persons for the 6-day short-term course on plant taxonomy. As Mathur sir rightly said, it is very, very essential to have a basic and fundamental science as a pillar of the house, as a foundation of the house, to endeavor in the allied branches, applied sciences, even including bioinformatics. We are very much delighted to have a guest like you, sir. I'm personally felt very happy because you did an MSc in zoology. I am also from the zoology from Osmania University. I felt very happy also. Coming to the lectures, we have arranged different areas, sir. We have arranged different kinds of lectures, different areas. And personally, I feel, and from the feedback, not today, for the last 6 days, every day after every session, we have sent a Google feedback form to each and every participant, and with certain questions of the particular lesson, particular talk. And the feedback also very nice. And so many teachers have answered questions or answered the question paper what we have sent to them. And in general also in the chat box, and in general also personally also they are making so many messages that they have got a very good knowledge, a very good information, very good subject from different participants, different resource persons, and definitely it is going to help them in a long way. With these few words, I think I will propose a vote of thanks to the valedictory session, and we personally will be in contact with you, sir, because you, as you said, we need support from the National Biodiversity Board and Regional Biodiversity Board to develop people's biodiversity registers and to do small projects with the help of our scholars, with the help of our students. So we will be approaching you very shortly with the our own this kind of idea, and any other help also we will be approaching to you very soon, sir. So with these few words, I'll thank each and everyone and who participated in the 6-day workshop, 6-day national short-term course, and all the dignitaries, all the resource persons who have given their valuable time to speak 1 hour, 1.5 hours, and entire their research work, their commendable research work shared with all the participants. So many resource persons have shared their PPTs including their valuable works, research papers, and everything will be shared with all the participants. With these few words, I thank once again all the participants, all the organizers from the bottom of my heart. Thank you very much.
Thank you very much, sir.
Thank you, madam.
Sir, I would like to say a few words.
Yes, madam, please.
Sir, on behalf of our organizing team, as well as the participant side, I would like to say thanks to our technical team, sir. Without this technical team, especially our scholars, Srujan, Sadhana, and Karthik, Ashok, Raju, all these technical team members, without their support, it was not at all possible to run this show, sir. So from all the organizers as well as the participants, I'd like to thank on this occasion the technical team to the maximum possible extent, sir. Thank you very much for giving me this opportunity.
Sadhana Vijayabhaskar Reddy, please.
Yeah, vote Sadhana.
Yes, sir.
Move to next part.
I would request the technical coordinator, Srujan, to give away his feedback about the session.
Thank you. It is a pleasure sharing my experience with you all again. First of all, I congratulate Dr. Vijayabhaskar Reddy sir, Dr. Ramanchi Nageshwar sir, Dr. Sasikala madam for the successful completion of the short-term course. And I also feel happy and glad to work with them. With the work, these 7 days work, I have learned so many things on part of the technical side. So I've exposed to many different new areas again. It is also a pleasure knowing all the wonderful sessions, very marvelous subject on screen. And off screen, we have been working on different areas. We have been preparing the question papers. We have been looking at the technical part. Now, I feel like I have become a cyber security expert or something like that. We have been putting down the hackers also. So I have been learning many things these days. So it is a great opportunity for developing such skills over a period of time. And without we getting a chance to do that, we will never expose it to certain areas. So this special short-term course has given me a chance to look into those areas, and I'm very thankful to the organizing team, organizing secretary, Dr. Vijayabhaskar Reddy sir, and my supervisor, Dr. Sasikala madam, Dr. Ramanchi Nageshwar Rao sir, and have been working with the same team. We have been organizing other courses, international seminars, e-conferences, and this is a short-term course. And next we are coming up with a new short-term course on green audit. So all these days, this is a wonderful coordination we have. A wonderful space I have got to work with. These are marvelous people. They are getting a lot of great personalities, great teachers, great professors from all over the world. I have been exposed to n number of international speakers, n number of them, the personalities, the great personalities in person. I was also given a chance to speak with them on certain occasions. So this has given me a lot of new enthusiasm. This has also given me more confidence. So with this, these kind of sessions, though the participants may be sitting from 10 a.m. to 4 p.m. every day, but the organizing team is day and night. We have been working on to make this show run successfully. So all of us, including our professors, our technical team, Sadhana, Karthik, we have been working late in the nights. I think all of you can also see us, even at night, even in the 1 o'clock night. If somebody is messaging, every one of us are replying. So that, I just want to make sure that, we have been working such a hard time. And by 6 a.m., I am on my desktop. So that is what we have been doing. That is the pain we are undergoing to conduct these online sessions. But this is a very pleasure moment for giving a course like this. Many feedbacks getting in a very positive sense. from great personalities, from people, not only just you are not just the participants, but you are very great intellectuals. There are many doctors, there are many professors in the participants list. We know that. So to get good feedback from you will always give us a great enthusiasm to move forward. So this is a pleasure moment for us. All our pains are gone in vain now. So very thankful to you all. Really thankful to everybody. Thank you, dear all participants, because your presence give us a very enthusiasm. So I should say that there are around 150 to 200 participants watching the session on live on Zoom, and there are around 70 to 80 live participants on YouTube, and there are around 250 participants watching YouTube by everyday night. If they are pausing at some point of time, they are coming back. They are coming back and watching the show again. So we have this. We have this statistics behind. We have been looking at the statistics, how the participants are participating, what is the behavior of participants, on what areas they are good at, on what areas they are enjoying the sessions. So we have a lot of things, from the this digital thing, we can get a lot of feedback, we can get a lot of things. So knowing all these things and understanding the dynamics of a short-term course, understanding the dynamics of a webinar, this is really a great opportunity for me. I really thank everybody. I really thank my professors, my supervisor. I thank everybody. Thank you. Thank you.
Yeah. Thank you, Srujan. Okay. No, I forgot to say thank you to all the technical team. Because you're all a part of the team. That is why I especially not remembered you, because we are going a lot of this thing these days, last 1 week onwards. So
Sir, it is not at all required, sir, because as a team, when we are working, the space you give us, that says a lot about anything, sir.
Yes, yes. Okay. Thank you. Thank you. Correct.
Yeah. Dr. GB is again joined.
Yes, sir. Yes, sir.
Yeah. We will continue the session. Please bear us, all the dear participants, half an hour more, because we ended the session unexpectedly. So Gibi sir joined again. We'll go with that session. Just half an hour, not more than half an hour, I think. Again, we'll come to the next part. Over to the Gibi sir. Sir, yeah. Please share your slides. Continue.
Vijay, we will take some more responses from the participants also, if they are interested. We'll
yes, yes. Okay, okay.
Every participant, please wait. We will complete the Gibi sir talk, and then we will continue. Anyway, as a formality, technically we have completed vote of this valedictory session, but we will continue this one presentation. Yeah. Please, sir.
So, Gibi sir.
Am I audible?
Yes, sir. Your voice is little low, sir.
Just a moment. I'll correct it.
Okay, sir. Okay, sir. Thank you, sir. We'll wait, sir.
How about the audio now?
Yes, sir. You're audible, sir.
It is very fine, sir.
Okay, just 2 minutes.
Okay, sir.
Okay, I'm sorry for the the interruption, and I'll continue wherever I stopped. So I think we were here, and yeah, we have reached here.
Sir, can you share your slides, sir?
Share your screen.
Oh, sorry. Yeah, is it coming now?
Yes, sir.
Yes, sir.
Okay. Yeah, we were here, and yeah. Okay, so yeah, no, we have gone even past. Posiflora also we have done. Yeah, I think we were here. So the next genus is Concidium. Concidium is a separate genus. Earlier, the species that is under the Concidium was placed under Area. So you might be feeling some similarities with whatever that I have discussed a little earlier with Area, the genus Area. So here, Concidium is a new genera, and there are a few species under the name or genera Concidium, in which we start with if the flowers are solitary, then it will be Concidium braccatum. Concidium braccatum. So it is very apparent. So there is not much complicated characters. So if it doesn't agree, then flowers more than one in raceme or spike, then you go to the next group. Then pseudobulbs more than 1 centimeter across. So you have to take a cross section along the width of the pseudobulb. And if you have the pseudobulbs more than, so the pseudobulbs are not visible from here, so then it will be, it become Concidium filiformis. Okay, then if the pseudobulbs are less than 1 centimeter across, then go to the next group of keys. Then if the scape is up to 1 centimeter long, then it will be Concidium microcheilos. And this is Concidium microcheilos. And also there is one more character which is not included in the key, is the zigzag arrangement of the inflorescence. And the flowers are very interesting and very photogenic, looks like a stellate shape. Yeah, I love this plant. Even though it is very small, it is very interesting. Then if the scape is up to 12 centimeter long, then it could be Concidium Nianam. Okay. So, I mean, we cannot compare the size because these two pictures have taken with two different lenses. Okay, then, yeah. So I was just introducing some keys, how to go about it and all. And there are even easier keys available for orchids. But there are even tougher ones also, like the smallest one, flowers of Oberonia is very tough. And you have to get clear characters of the lip, then only you can identify those orchids. So I've been working in the Western Ghats for the past 15, 20 years, and I have developed an interest in orchids along with other groups, also along with the reproductive biology. So I have collected a number of orchids from the Western Ghats. We have about 300 species in Kerala. It's in the Western Ghats, about 340 species are there. So I could collect some. So I've categorized it into it accordingly, according to their nature or habit. I mean, epiphytes, terrestrial, and saprophytes. So some of the epiphytic ones, I have selected some which are rare. Okay, so this is the Diploprora championi, and this particular plant was not been reported from the Western Ghats until 1975, I guess, when Dr. Father Saldanha, who wrote complete flora of has reported this from the Hasan district. In his flora of Hasan, he had described this particular plant, and it is very difficult to locate because it will be growing among the branches of small herbaceous plants or small trees, and the leaves are very difficult to distinguish from the what is a horse tree. Okay, and this is a wonderful flower. Even though the flowers are a little smaller, like 1.2 to 1.8 centimeters across. So even though they are smaller, they are very photogenic. Okay, and another group is Gastrocytelis, and I'm thankful to TSP Komasar, who was a forest conservator in Karnataka. He had sent lots of pictures from Karnataka to eFlora of India, and I'm using his picture to explain some of the one, and I'm using some other pictures also that are taken from different people. So Gastrocytelis acalis, and it is also the flower is also very interesting, and yeah, there was one more picture that is not there. Okay, then another one is Smithsonian Maculata. The name itself is very pretty. So this is an evergreen species. You can see the background is very dark. This is an evergreen species. Again, TSP Komasar has taken this picture. from Karnataka, central Karnataka, and the flowers are very pretty. Yeah, Smithsonia, then Smithsonia viridiflora. So here the flower, the Smithsonia Maculata, the flower is like this with some blotches on the petals and sepals. Yeah, but when you come to Smithsonia viridiflora, the leaves are little smaller, but the inflorescence are tufted, like a tuft or group, and flowers are very pretty. And when you take a single flower, this is a single flower of Smithsonia viridiflora. This I have taken from Kudremukh National Park. Okay, then Dendrobium nodosum, earlier name was Flickingeria nodosa. Now it comes under Dendrobium nodosa, and because of this character, it has got the name nodosum, and the flower is very pretty. I have shown you this the very same picture earlier. Okay, then Oberonia Chandrasekharanai or Oberonia Rengannayana. There are a few synonyms for this particular plant because different people have identified and reported this plant from different places. Anyway, it is a very interesting plant and it flowers in the rainy season. You can see that the wet I think this is the time for flowering of this particular plant. And it's very nice. The entire the flower, what do you say? The entire inflorescence is with open flowers, and this will continue for a few days. The flower lasts for a few days. And you can imagine that this particular flower might have started opening, or it must have opened a few days or a few weeks back. I mean, and it was waiting till the what do you say? The new buds to open. And it may stay back for some time to maximize the opportunity for getting pollinated. And look at the flowers, I mean, 1 to 2 millimeter sized flowers. Another flower, probably one of the smallest flowers from the Western Ghats. And just a moment, I'm sorry that. Yeah, probably one of the smallest flowers of the Western Ghats, which is about little less than 1 millimeter. I have seen only one inflorescence with flowers of 1.2 millimeters. And it was also a little tough to take a macro picture. And yeah, so this is the picture. This is the what is a macro picture of what is I've used 105 mm Nikon macro. And yeah, Oberonia tenuis. It is very interesting, and also, I mean, nobody knows what kind of function, what kind of pollination happens, and the fruit set is very, very less. For an inflorescence like this, I could see only 1 or 2 fruits that is developing, and it is extremely rare. Only in the mid altitudes of Karnataka, Kerala, and Goa, Maharashtra region. And there is another plant. Unfortunately, I don't have a flower picture of Porpax jordoniana. This is a rare endemic and threatened plant. Okay, another one, Sudan Ferdinand Filiformis, very pretty orchid, which is reported from Kerala, and I think it is now reported. I mean, it is now growing or seen in Kerala, Karnataka, and Tamil Nadu. Okay, and very pretty flowers, lovely flowers. Okay, another one is Rubicusia josephiana, and there might be there is a debate over the name of this particular plant. I have identified this as Rubicusia josephiana. Maybe there is a change in name or a difference in name. But this is also, I mean, the flower is little smaller, but and also this particular plant is growing sometimes in the canopy, sometimes in the lower branches of small trees. But it is a treat to the eyes in getting this plant. Okay, another, I mean, probably the smallest flower among orchids. I've shown you the flower of this particular plant, Succulebium gemmatum, with measurement 1.5 millimeter or lesser than that. And the size of the flower ranges from 0.6 millimeter to 1.5 millimeters. Okay, and this is the very tight close-up and cropped picture of Succulebium gemmatum. Okay, another very common plant, Chrysostoma tenuifolium. It's very common in the western slope of the Western Ghats up to mid elevation, and very tiny, but the coloration and the brightness may vary from place to place. Okay, then Pholidota pallida, but I've shown you the plant when I wanted to describe or tell you about the pseudo bulb, but I haven't shown you the flowers. This is also a very tight close-up and cropped picture, and you can see that ants are moving around. Okay, but we haven't seen any ants taking the Polynia away. Okay, so these flowers are little tiny, but very interested, very interestingly arranged floral structures. And there is another plant which is Polystachya concreta. Polystachya concreta. And there is one more plant you can see, that is a bulbophyllum. Okay, so but I have placed this particular plant or flower here to show you that there are orchid flowers which will not follow the resupination, or it may be following 360-degree resupination. But here look at the this thing, what is the buds, the smaller buds also having the orientation like this. So the lip is placing upward. So someone has to find out what is the functional purpose, why these flowers are are having such kind of differences in what do you say, their approach or strategies. Another interesting plant is Xenico Phytons Nianam. The name itself is very very stylish. Xenico Phytons Nianam. This is also an Asian plant, a tropical Asian plant, which we found from the grassland mixed with the shola forest. Okay, and flowers are very pretty, just like someone taking their tongue out. Yeah. Okay, then some of the terrestrial orchids. This is Habaneria diphylla. And yeah, this is the picture of flowers of Habaneria diphylla, just like horns of buffalo. And then Habaneria crenifera, we can call it as South Indian dancing beauty or dancing girl or something like that. Yeah, and another one, Habaneria longicorniculata. You look at the spur. And if someone knows the history of Darwin orchid and Angraecum sesquipedale, and he predicted the pollinator during 1630s or 1640s, and he was saying that considering the length of the spur, the length of that spur was the Angraecum sesquipedale is about 29 centimeter. Here it is about 10 to 15 centimeters. So what he predicted was that considering the length of the spur and the color of the petals, white petals, then the pollinator could be a moth which is active in night, having a proboscis length as much as that of Angraecum sesquipedale, that is 29 centimeters. So here that prediction is very valid. So this also, considering that the white color and the length of the petal, so you have to expect some, what do you say, a nocturnal moth which is having a proboscis length of about 10 to 15 centimeter. Thanks, Dr. Satish Patke, for this wonderful picture. And yeah, this also. And you see the Polynia comes out, which is little blackish in color. And looking at this from this angle, you may feel that it is mimicking something, some structure, I don't know what. Okay. Yeah, then another one which is very related, even the name is also very related, Habaneria longicornu. The leaf shape is different. The length of the, what do you say, the spur is little shorter than that of longicorniculata. And this is mostly seen mixed with grasses in grasslands. Okay, so longicorn, even the lip is also a little different with the broader sepals. And you may be able to see the nectar which is filled inside the spur somewhere here. No, no, it is not demarcated here. So you could see nectar filled inside the spur in such species if you happen to see this in the field. And another one which is again having very small flowers, Dendrobium herbaceum. This is very small flowers, but when in flower, very few leaves, and the flowers are quite large number of flowers, and these flowers are fragrant. These flowers are fragrant. Some of the orchid flowers are fragrant, but you may not get it from a distance. So wild flowers, when you see, then you have to smell it. No harm, but only allergic people have to taken care of. Otherwise, even though they are very faint smell, they are very pleasing smells, and you might not have got that kind of very unique smells from orchids, wild orchids, but they are very, very minute. Okay, another one, Nervilia prainiana, very interesting flower, but when you look it from a distance, you may not be able to see this beauty. But I mean, and also this will last only for one day, one flower per plant, and it will appear immediately after the summer showers, before the monsoon starts. And this is very pretty on a closer look, but it is very hard to locate. Okay, because very small. The entire inflorescence itself is about 6 to 8 centimeters long, and the flower is about 2 centimeters or less than 2 centimeters across. And another one which is very pretty, and as you can see the background, this is taken from Bababudan Hills in Chikmagalur district. And Satyrium nepalens, it is very, very crucial and important as far as biogeography of plants is concerned, because it do have a very disjunct distribution. Yeah, the flower is very interesting, like an inflated balloon-like structure with the upper petals, the upper petals. Okay, then another interesting terrestrial orchid which is seen inside the forest, mostly in the tropical evergreen and semi-evergreen forest, which is called the Zeuxin longilabris. Zeuxin longilabris. Thank you, Mrs. Smita Raskar from Pune, for giving this wonderful picture. And yeah, Zeuxin longilabris, nice flowers, very, very pretty, pure white colored flowers. Again, very tiny flowers. Again, another 1.5 millimeter, or even sometimes some of the flowers may be 1 millimeter. Heterotrophic parvifolia. This is a what do you say, lithophytic orchid seen growing in on rocks of evergreen forest. That too in the mid elevation from about 600 meter above. Okay. Then saprophytic orchid. This is Epipogeum roseum. Epipogeum roseum might be there in most of the mid elevation region by now. Or there is no specific time period for this to flower. It depends upon the moisture content of the topsoil and the shade. Then it might be depending upon the summer showers or the rainfall. During heavy rainfall, when the floor is very heavily wet, then this particular orchid might not be there. Okay, then this is the flower picture, and you can see that the roseum, Epipogeum roseum, rose-colored blotches, but sometime you may not be able to see those blotches on the petals. On a white background, if you see such blotches, such very contrasting blotches, that is very interesting. And there are so many. There are so many. If you go to places like this in the Western Ghats, this picture I have taken from the Southern Western Ghats from Agastyamalai, and a place like this in Silent Valley, like a moist place, which is very much necessary for orchids to maintain its life history traits. Moisture is so humidity is one of the most important factor, at least for the epiphytic orchids. And then typical habitat, there are differences. You may be able to see some group of orchids, some species of orchids on the tree trunk, some may be on the primary branches, but this may be different. The one that you see on the main trunk might not be distributed in the, this bifurcations or the primary branches or the crown area. So different places, there is a spatial distribution within a microhabitat. If you consider a tree as a microhabitat, within a tree itself, a tree trunk may have a group, and the primary branches may have a different group. And then when you go to the leafy vegetation, you may see very different orchids. And among the leafy vegetation in the canopy, you might see very, very tiny orchids like the one that I've shown you earlier, which is leafless, Geniophila. Okay, then branches, which is open, you might be able to see orchids and other pictures. This is not from in India because bromeliads are there. So this is not from from India. Okay, and also they are very much related to certain other plants, which I mean, at least some orchids are very much associated with bryophytes, especially, or pteridophytes. And we are trying to understand what are all the specificities of the orchids with their associating plants. A work is going on to identify what are all the probable what do you say, associated plants, bryophytes, pteridophytes, even some other epiphytic angiosperms. And also I have studied the what do you say, other parameters, ecological parameters like light, shades, then slope of the area, then the tree height, things like that. So yeah, so there are so many other parameters, what do you say, affecting orchid diversity. So you, and also another important thing that all over the world, including I have studied this particular point, that only primary trees, only the larger trees with a huge trunk size, with larger, what do you say, GBH, only supporting orchids in tropical forests. It may be different in Eastern Ghats because there the tree trunk or the tree girth is little smaller than that of the Western Ghat area. So in Eastern Ghats, in your area in Hyderabad and all, you may be able to see orchids on smaller trees also. But in our area, it is the picture is very different. In smaller trees or in a secondary forest, it is very different. A very primary forest wherein larger trees are there, then only you would be able to find orchids. There are few orchids in on this particular tree, along with this is one species. This is Area posiflora. There is another Dendrobium, and there is one Bulbophyllum, and there are few other area where on this particular tree. So on a single tree, you might be able to see several species, and here also Area posiflora and other orchids are there. Okay, and this is the major threat for orchid diversity. Fragmentation and deforestation, and also encroachment in places which is, I mean, we need to have fragile, I mean, very intact forest which is not disturbed. In disturbed forest area, epiphytic diversity is very less. If it is there, I mean, the species which are distributed in distributed habitats will have high resistance towards opening of light, opening of forests, gap formation, things like that. And which are susceptible for any kind of such damage will succumb to death and they will vanish. Okay, so what you can do is that you can rely upon certain sites. And I would suggest you to go for eFlora of India. About 1,200 entries are there so far. And there are lots of information available. So you just go and search Orchidaceae, and then there are even including discussion you will get. Okay, and if you have some orchid for identification, you can send it to eFlora of India. All you want to do is that you have to become a member of eFlora of India, and then you can send your pictures. There are lots of experts. I'm also fortunate to be a moderator of this particular group, thanks to J.M. Garg, Gurjaran Singh, and the like greats. Okay, so this is one side you can rely upon because there are so many experts. Even Dr. Pankaj Kumar, Jivan Jalal, all those orchid experts are there. So you can go for. So nowadays there is a huge demand for online image search. You just look at one image that I have uploaded today morning. Today morning by around 11 o'clock, I have uploaded one picture. I knew this plant and I wanted to know the accuracy of identification of image identification. And then they have identified it as a lady slipper orchid to me. This is, whereas this is a Malaxis reedy or Malaxis. So, so don't go for, at least until the algorithm working behind such kind of sites perfect enough to give you identification. But I mean, it might work for horticultural plant or common plants that are distributed in urban lands or in our land. But don't go and identify wild species with such kind of any sites or even apps. Okay, so can you see something? I mean, the what do you say? The surprise that orchids are keeping for you is very different. You can see something vague, no? This is also an orchid and it is called the Dracula orchid, as you can see the color. So if you haven't seen black colored flower, here it is, a Dracula orchid. Okay, yeah, then some significant references for orchids is the recent publication 2019 Orchids of India by a pictorial guide by Botanical Survey of India. I'm sorry, I haven't got a copy yet, but I have seen some inside of pages from my friend Dr. Jivan Jalal. Okay, then Orchids of Eastern Ghats. Yeah, it is also, this would be very helpful for people from the Eastern Ghats like Hyderabad or Visakhapatnam and places like that. Then, yeah, this is a classic book for Indian orchids. Indian orchids, the volume 1, I've placed only Uday C. Pradhan. Wonderful key, but the only problem is that this is published long back. After the publication of this particular book, there are lots of updations happened. So one has to update oneself or yourself to know that what are all the updations happened in that particular field. So now I hope that there are so many people working on monographs, and I hope that soon monographs of orchids will publish by different authors. Then Lady Slipper Orchids of India, H.J. Chowdhury. I have seen it when I visited Meghalaya, and it is wonderful, very nice pictures. And you can go for if you love Lady Slipper orchids. Then Orchids of Maharashtra for Western Ghats, at least Northeastern Western Ghats. Again, Botanical Survey of India published this book. Then 100 Northeastern Orchids by Mohan Pradhan. Again, the pictures are wonderful in this book. Then Wild Orchids in Karnataka, a pictorial compendium. Again, this book, I've referred this book. The only problem is that there are a few names have changed, and when you run keys of certain genera, you may feel it difficult because some updations have happened, and unfortunately the team behind the T.N. Anandarao sir and S. Sridhar hasn't incorporated those changes, but otherwise the pictures and the drawings are wonderful, and also explanations or descriptions of plants is wonderful. And also the introductory part, even the previous version, Wild Orchids of Coorg, is also very interesting, at least for a beginner to have some introductory understanding. Then these books, and also this Introduction to Orchids by Abraham Valsala, although this is mostly for Kerala orchids, but the introductory part covers a very good introduction of orchids in general. And also this book, Introduction to Orchids, deals with the horticultural aspect also. Okay, and I would like to thank Dr. Jivan Jalal and Pankaj Kumar, orchid experts in India and worldwide. Dr. Pankaj is now working in Hong Kong Botanical Garden, and he's an interesting person, these two. And also I would like Flora of India for lively interactions, discussions, and accommodating me. Thank you very much. Thank you very much. You can contact me at gbykuriakos@shcollege.ac.in. giby.kuriakoc@shcollege.ac.in. Thank you very much. And I would like to place on record my sincere thanks to the organizers. Thank you.
Yeah. Thank you so much, sir. Thank you so much. Actually, we would like to end course with the in a pleasant way. So we thought yours is the best one for this. So we
thank you so much.
Yeah, it is according to our expectations. I hope our audience also has same feeling. And you presented many of the orchids and their taxonomy that enriched many of our participants' knowledge. We have very few orchids are in Telangana.
Yeah, I know.
Yeah. For any further help or visit when we come there, we would like to contact you.
No problem. You're
Yeah,
welcome.
yeah.
You're welcome.
Yeah, thank you so much. And some of the participants would like to interact with you.
Sure. They can.
Sadhana?
Yeah.
Them to raise.
If there is any, I mean, if you have chat box and if there is any chat and questions, please. What is one question? What is the meaning of orchid? Unfortunately, I don't know. I'm sorry, I don't know why I haven't. So far, I haven't keen in knowing the what do you say, the meaning of orchid. I'm sorry. And certainly, I will get back to the person. You can please what do you say, note down the email ID of that particular person. I will get back to you.
Sir, from my side, one question, sir. Among the terrestrial orchids, can we propagate the orchids in tissue culture lab or any other method so that we can explore? Are there any efforts from the scientists or government of India to explore different kinds of terrestrial orchids to make them as ornamental outside?
That's a very valid question, because so far lots of people are interested in bringing up epiphytic orchids as horticultural significance. So I will tell you why I mean terrestrial orchids are not being considered, but there are terrestrial orchids. The lady slipper orchid is a terrestrial one, and then Arundina graminifolia is a terrestrial orchid. Okay, so there are so many terrestrial orchids which are of horticultural value and you can cultivate it easily. But the problem with cultivating or domesticating wild orchid is that, I mean, they are very difficult to domesticate, because I mean, might be because of the clear-cut soil characteristics. And no one knows, I mean, very few people have worked on what are all the parameters that influence orchids in general, whether it is epiphytic. I mean, some of the epiphytic orchids I tried several times, and I tried bringing back to its own, I mean, analogous habitat places provided enough, what do you say, background or habitat, but some orchid couldn't thrive. So we don't know what are all the ecological, climatic or other factors which are governing or which are controlling these orchids. So for terrestrial orchids, soil is the most important thing. And the majority of the terrestrial orchids are in constant communication with the mycorrhizae. Okay, so if you are bringing these rhizomes or terrestrial orchids into a place where this mycorrhizae cannot establish, or the mycorrhizae is not at all there, then it would be very difficult. So we have to have, I mean, I'm trying for a study to understand what are all the parameters, that is ecological parameters or soil parameters, that are going to affect terrestrial orchids. So unless we have such studies, it will be very difficult to explain why it is not. And for this thing, biotechnology or tissue culture purpose, I mean, they can easily what do you say, propagate by means of their rhizomes or tubers. But but I don't know whether we can establish there in a condition that we are providing. The problem that I've told you, that the mycorrhizae could be probably the mycorrhizae, lack of mycorrhizae. Another thing is that for terrestrial orchid, the fruit set is very, very low. For about 20-30 flowers, you might see only 1 or 2 fruits. Sometimes an inflorescence of 20 to 30, there won't be any seeds. From seeds, you can bring, you can easily germinate in what you say, under lab conditions. But the problem is that when you want to, what do you say, culture it, harden it and culture it outside the lab, then again, this problem might come up. That was a nice question.
Thank you. Thank you, sir.
Oh, Shiv, Needle, Shiv, you are there. Thank you.
So some participant named Srinivas Reddy.
Okay, yeah, please.
Sir, please pose your questions.
Srinivas Reddy, sir.
Maheshwari Koti.
Yes,
Madam.
yes, sir. Am I audible, sir?
Yeah, yeah.
Yes.
Sir, I belong to the eastern part of Karnataka, where we have the scrub jungle of forest variety. Sir, is there any possibility of orchid in our district, Bagalkot, sir?
Bagalkot, where is it? What do you say? Considering Chikmagalur?
No, sir. It is in eastern part of Karnataka.
Okay, so maybe
Earlier the
towards.
district, sir. Earlier the district was belonging to Vijayapura district.
Oh, okay, okay. Then it is an arid zone with very less rainfall.
Yes, sir. Yes, sir.
Okay, so I think it would be very difficult. But in your area, there are few terrestrial orchids, like
Yes,
some
sir.
Habaneria and all. We have seen Habaneria roxburghiana and all in such places. In Bangalore and all, when I was working in Bangalore, we tried several times and it was not successful. But garden varieties you can easily cultivate in a greenhouse. You can keep it in a greenhouse. But wild orchids, it's very, very difficult.
Yes, sir. Okay. Thank you, sir.
Okay, there is a good question from I don't know how to spell it. Nartana. Do you think this COVID period, which bring back many endangered species, extinct orchid? No, fortunately, we don't have any extinct orchid as of now. But there are so many orchids which are on the verge of extinction or very threatened condition. And also, we can vaguely say that, yeah, nobody is going inside the forest or no harm is happening to the wild and the climate is very clear. Nowadays, pollution rate is very low. But if you want to scientifically speak, you have to have data. Without having any precise data and say that COVID will bring back some species, then it would be just a general statement. But that is as of now, it can be a very good belief, and also you can see that or think that it would be a reality. And also, I would assume that we may have a better tuning to understand that these are all necessary things. So when we are sitting back at home and when we are seeing these problems that is happening outside, and we can easily, or people can easily realize the fact that there are so many unknown treasures there in the field, there in the wild, which we don't know what is it for. And there are so many orchids which are being utilized for medicinal purpose, and even now we don't know which are all them. Some of the Habaneria is being utilized, and there are few works from Uttaranchal and a few works from Sikkim, then some work from Kerala regarding the medicinal property of orchids. And there are some orchids which do have very specific utilities. So I hope it will work out. Okay.
Srinivas Reddy, sir, please pose your question. Srinivas Reddy.
You can write, sir, Srinivas sir, if your audio is not working, you can write it down in the chat room.
Sir, somebody is asking edible orchids. Are there any edible orchids?
For me, I don't think so. I haven't come across any edible orchid, but there is no poisonous orchids. Maybe some of the rhizomes, there may be some elements, but leaves. I heard that in northeastern, some of the northeastern states, the leaf is used as vegetable. I don't know which leaf. And there is another question. Venkadesha Ram, is there any relation epiphytic orchid with the host? That is a, I mean, that is a question. Since I have worked on it, it is a, what do you say? I may need to give a very broad answer, but I will conclude it in a few sentences. That there are quite a lot of studies across the world to understand that whether there is any species specificity of epiphytic orchids with respect to their host, and found that except very few orchids, that orchids in general do not have any host specificity as far as the host species is concerned. But yes, there is a specificity towards the bark characteristics and also the trunk nature of the architecture of the tree and type of foliage and the depth of the foliage, and then the dispersal of the branching, like how spread out the branching and things like that. So there are more of micro habitat conditions than that of species of the orchid, which is specific. Why charcoal is used in cultivating orchids? Any plant species association of ground orchids? Yeah, there are so many associations of ground orchids. Sometimes, as I've already mentioned, that there might be mycorrhizal fungi, and also there may be some other species, because epiphytic, terrestrial orchids are not growing alone. They are growing along with grasses, other plants, and some other soil microbes, flora and fauna. So there may be association, but so far there are no much studies to understand these questions or to answer these questions in detail. But we do have qualitative information that that states that, yeah, this particular orchid is seen growing always along with this particular species of grass, or this particular species of herbaceous plant, and things like that. And charcoal is a very good substratum, and it is not only a substratum, it provides enough minerals and nutrients in the required amount for an orchid, because it doesn't require concentrated fertilizer or concentrated nutrients. So charcoal is giving enough, but it's very specific. Yeah, I already told that there are no poisonous orchid as far as I know as of now. Chandra Prakash Kodam was asking a question. Yeah, there are some rhizomatic rhizomes that might be poisonous if consumed in a high quantity, but there are no records or there are no evidences so far to state that the orchid, any part of orchid is poisonous. Okay, nice interaction also. Yeah. If there is any other questions, or if there is anything comes to you, you may please direct it to me. And thank you for the surprise invitation, and also thank you for giving me 2 chances for a 1 hour lecture. Thank you very much.
Thanks a lot, sir. Thanks a lot. You showered us with a variety of orchids. Now over to Dr. Amanchi Nageswara to propose a formal vote of thanks.
Yeah, it's my duty to propose vote of thanks from the organizers side. On behalf of the entire organizing committee and on behalf of myself and the participants, we are very much thankful to you, sir, Gibi sir, for readily accepting our invitation and giving a very good lecture, very good talk, and definitely it inspired so many participants with lot of information, with lot of details and different types of pictorial this thing. Definitely, I hope that we have given very good opportunity to all the participants to listen your very good lecture, informative lecture. So with this, I'm very much thankful to you once again.
Thank you.
And we hope my colleague Dr. Vijay Bhaskar Reddy will be contacting you further in future also.
Thank you. Thank you.
Yes,
You're
yes, sir.
always welcome.
We have one Habaneria. We'll come to you, sir, with that.
Sure, sure.
Detail.
Sure.
Okay. Thank you, sir.
Thank you.
Thank you. Thank you very much, sir. Yeah. Any participants would like to interact or say something? Before concluding the entire 6-day session, Srijan will make some of the any announcements are there, and everybody will get a certificate without fail, but based on your attendance and feedback only. Those who have attended complete 6 days with all kind of your feedbacks and assignments, those will get the certificate. If anybody is not having details or anybody is missing with the details, please let us know, so that please send your details, participation details, so that we will try to give certificate also. Yeah, and any hardships or any shortcomings from our side, we are sorry. But as our organizers, we have to make sure that it should be a strict program. That is why we have given all the assignments, all the questions, and we have tried our best to give best lectures and best times to spend with us all these 6 days. We hope you understand all the difficulties, and we have also learned so much of so many things, so much of knowledge from these 6 days course. Especially, I'm very much personally thankful to Vijay Bhaskar Reddy because as a team we are working, but since he gathered so many resource persons of different disciplines, and it is also technically very useful to me. I have been in the last 1 week, I have been watching all the lectures. Few of them are very much interested to me also, but unfortunately, I could not do any work in this area. But for that, I'll try to have some particular collaboration with all the participants, with all the resource persons. With these few words, I thank you one and all. Over to Vijay Bhaskar Reddy.
Yeah, thank you, Dr. Ramanji. Actually, I would thought of offline a short-term course, but due to the COVID, that was postponed, and we got a good platform. So because of COVID and because of technology, we all are connected today. And because of strong technical support from Srijan, Karthik, and Sadhana, and my co-organizers, Dr. Amanchi and Dr. Shashikala, with good coordination, we wind up all the sessions very successfully. And today we are at the end of the 6-day course. I thank each and every participant for your patience listening. And my side, actually, I'm a palynologist. I would like to present a few of my research work also, findings also. But because of the time, I couldn't. Anyhow, this is a platform where I met very eminent personalities and many scholars, teachers, professors from different parts of the India. We'll continue this rapport, and already we prepared that PDF file, all the participant list. Please be in touch. Any problem in identification of the plant species, we can cooperate with each other. And actually, taxonomists are rare, endangered. But many of taxonomists, because of the some ego, they are becoming isolated. We should not have that ego. We should leave our ego and work with each other. Then only we can raise this subject taxonomy to the upper heights. That's my personal feeling. And also, we invited Dr. Madhur for this session, valedictory session, because he's a chairman for the National Biodiversity Board. Each and every state is having the Biodiversity Board. But what happened in Biodiversity Board, first initial state, they're approaching the taxonomists to compile the data from one area. And later that depending on their own people, and they're not giving any funding to the universities or teachers. That's a problem with the biodiversity boards. So from your side also, please bring it to the notice of the National Biodiversity Board Chairman. And no other intention in this. Only thing is for the welfare of the society. In that motto only, each and every person, taxonomist must work with the biodiversity board and strengthen the biodiversity board and strengthen the local activities so that it helps the national flora. We can protect the endangered, endemic, rare, threatened plants. And with this, I conclude, and a big thank to each and everyone who supported for this 6-day short-term course, because we faced many problems from our side. Now those problems, by seeing you all with the number more than 150, 200, we forget all the pains in arranging these sessions. You all made us happy. You made us happy to organize this event. So big thank to all the participants, and don't be hurry in getting the certificate. Give us time, because tomorrow onwards we are starting one more program. Already we exhausted, but we are putting some energy for next session also. So don't be hurry in getting certificate. Give us time up to 25th August. Once everyone gets a certificate, and we'll wind up the groups also. Don't worry, and don't pour any mails on our technical team. Already they are exhausted, and please be patient and fill that form which Srijan already circulated. With this, Madam, shall we end the session?
Yes, sir.
Yeah.
It's very happy and very energetic, sir. By seeing even now also, there are 1 or 3 participants.
Yes, yes.
1 or 3 participants. Actually, technically speaking, vote of thanks, valedictory function was over. But it is a rare case and very strange case that the speaker, valuable speaker, again coming and giving a lecture, continued the lecture. This is a very great thing, sir. I have never seen such a program, and the interest with which the speaker, Gibi sir, gave, it was very worthy noting, sir. Everyone should learn this, how he has interestingly gave the lecture, and how he inspired the participants. It was worthy noting, sir. Thank you very much, sir, Gibi sir, and also big thanks to all the participants. And as just now sir said that again from tomorrow onwards we are going to start the same team. We are going to start another program. So please forgive us or excuse us for getting certificates. It may be delayed because from tomorrow onwards our what we call works will be different. So there is no point that we are not going to give you certificate. Definitely your certificates will be given, but please give us some time, because every one of us are exhausted already, but again we have to gain energy to do next program. So that's why please excuse us. Definitely you will be getting certificates. We will be giving the certificates, but it may take some time. With this note, thank you very much, one and all. Thank you very much, sir.
Thank you, madam. We will wind up the session with the national anthem. Over to Srijan. Dear participants, please stand up for the national anthem.
Yeah, dear participants, please stand up. Even the panel, you have to stand up. I'm going to share my screen and the audio too.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल वंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you.
Thank you all. We are ending the session.
Thank you. Thank you, one and all.
Thank you all participants. Have a good day. Bye.